could be exploited in your future programs. So, Andres, you're most welcome to just start your lecture.
Thank you very much, and good morning. Good morning, everyone. On my next lecture, on which I would like to present key methods used to analyze plant stress responses at biochemical, molecular, and imaging levels. I would like to explore advanced technique available by assessing plant stress markers under heavy metal exposure. Of course, I would like to begin with an overview of the biochemical and physiological assays that measure oxidative stress and antioxidant response. I would like to discuss spectroscopic and chromatographic techniques which can help identify metal accumulation and phytohormonal changes. However, about detection of isolation, extraction, and detection of phytohormone, I told you in previous lectures. Let's begin with the introduction of the adverse effects of heavy metals on plants. Heavy metals contamination in the environment is a major threat to plant health and agricultural productivity. Elements such as cadmium, lead, mercury, arsenic, chromium, and nickel accumulate in soil due to industrial activities, mining operations, and excessive use of chemical fertilizers. Heavy metal stress disturb multiple cellular processes leading to physiological and biochemical damage. One of the most critical consequences of heavy metal exposure is, of course, oxidative stress. Heavy metal interfere with electron transport in the mitochondria and the chloroplast, causing excessive production of reactive oxygen species. These highly reactive molecules damage cellular membranes, proteins, DNA, leading to cellular dysfunction and ultimately cell death. Another significant impact is photosynthetic inhibition. Heavy metals damage photosystem II, which plays a vital role in light-dependent reaction on photosynthesis. On the previous lecture, Professor Shamsul discussed about the role of phytohormones, the role of brassinosteroids on photosynthesis, and told about the fundamental role of photosynthesis in plants. However, in the results when the plant are treated with heavy metals is a decline in chlorophyll contents and reduced energy production, severely affected plant growth and development. Moreover, heavy metals induce a nutrient imbalance by interfering with the uptake of essential elements such as calcium, magnesium, zinc, or iron. These nutrients, of course, are very important elements for enzymatic reactions, signal transduction, and overall plant metabolism. When they absorb in hindered, key physiological processes, including protein photosynthesis or enzyme function, became compromised. Additionally, heavy metals can directly interact with DNA and proteins, binding to sulfhydric thiols groups of enzymes and structural proteins, and this disturb gene expression, enzyme activities, and metabolic pathways, further intensifying the stress response in plants. As a result of this disturbance, plants struggle to maintain normal growth and survival in contaminated environments. To better understand this effect, we can employ various biochemical spectroscopic and molecular techniques to analyze stress markers and evaluate plant responses to heavy metals. In addition to the oxidative stress and the nutrients imbalance, heavy metals toxicity significantly affects plant hormones, of course, which are critical regulators of growth, development, and stress adaptation. We know everything about action of phytohormones and classes and signal transductions and biochemical action in plants. However, for example, auxin is often reduced under heavy metal stress, since auxin regulate root and shoot development. Their decline leads to inhibited root elongation, stunted growth, and impaired cell division. Consequently, abscisic acid, as we know as a stress response hormone, is typically upregulated in response to metal exposure. Abscisic acid helps plant cope with stress to closing stomata, to reduce water loss and triggering defense mechanism. However, excessive ABA accumulation can lead to growth retardation and reduce photosynthesis. Similarly, gibberellins, which promote stem elongation and seed germination, are often down-regulated under stress, leading to reduced growth and developmental delay. On the other hand, jasmonic acid and salicylic acid, which are stress-related hormone, increase under metal toxicity. This hormone activate plant defense responses, including antioxidant production and metal detoxification mechanism. Understanding how heavy metals influence these hormonal pathways is essential for assessing plant stress responses and adaptation strategies. So one of the primary indicators of oxidative stress is the accumulation of reactive oxygen species, such as superoxide radicals and hydrogen peroxide. These molecules damage lipid, proteins, and DNA, leading to impaired cellular function. To counteract oxidative stress, a plant activates the antioxidant defense system, which include both enzymatic and non-enzymatic antioxidant. Enzymes such as superoxide dismutase, catalase, and peroxidase play a critical role in neutralizing reactive oxygen species. Meanwhile, non-enzymatic antioxidants like glutathione, ascorbic acid, and phytogelatin help detoxify heavy metals and maintain cellular redox balance. One of the most significant biochemical markers of oxidative stress is lipid peroxidation. This process occurs when reactive oxygen species attack polyunsaturated fatty acids in the cell membranes, leading to structural damage and loss of membrane integrity. The primary byproduct of lipid peroxidation is malondialdehyde, MDA. It serves as a reliable indicator of oxidative stress levels in plants exposed to heavy metals. So how does this process occur? Under metal stress, reactive oxygen species such as superoxide, hydrogen peroxide, and hydroxyl radicals accumulate in plant cell. These reactive oxygen species react with membrane lipids, initiating a chain reaction that leads to the breakdown of lipid molecules and the formation of malondialdehyde. The significance of MDA accumulation lies in its direct correlation with membrane damage. Higher MDA levels indicate greater oxidative damage resulting in disturbed cellular function, impaired water and nutrient transport, and ultimately reduce plant growth. To quantify MDA level, we can use the thiobarbituric acid test. In this assay, MDA react with thiobarbituric acid to form a pink color complex, which can measure using spectrophotometers at 532 nanometers. This method provides a precise and reliable measure of lipid peroxidation in stressed plants. This is the first method. Spectrophotometrically, we can measure stress markers. The first stress markers is MDA thiobarbituric, MDA malondialdehyde. In addition to lipid peroxidation, another important indicator of cellular damage under heavy metal stress is electrolyte leakage. This test assays the integrity of plant cells membrane by measuring the count of ions such as potassium, calcium, and chloride that leak out of the damaged cell. Under normal conditions, plant cell membrane as a selective barriers regulated ion transport and maintaining cellular homeostasis. However, when plants are exposed to heavy metal, this toxic ions disturb membrane stability, causing structural damage and increased permeability. As a result, essential ions leak out of the cell, indicating loss of the membrane function. So, how do we measure electrolyte leakage? The electrolytic conductivity method is commonly, of course, used. In this assay, no. Where's in this assay, leaf discs of root segmentation are collected from the plant. The sample are incubated in distilled water, allowing the ions to diffuse out. The electrical conductivity of the solution is measured before and after autoclaving. Autoclaving completely disturbs the cell membrane, releasing all intracellular ions. The percentage of electrolytic leakage is then calculated as an indicator of membrane damage. Higher electrolyte leakage values correspond to the greater cell membrane injury, reflecting severe heavy metals toxicity. This test provides a rapid and effective way to access early sign of stress, helping us to evaluate plant tolerance and tendency to metal contamination. By combining electrolyte leakage measurements with lipid peroxidation assay, we can gain a comprehensive understanding of how heavy metals impact plant health at the cellular level. Reactive oxygen species, one of the most, maybe most significant produced in plants, superoxide. This highly reactive molecules is primarily formed in the mitochondria and the chloroplast when heavy metals disturb electron transport processes. How is superoxide generated? It occurs when molecular oxygen gains an extra electron, often due to the interference of metals with electron transport chain. The reaction is follows. Another major source of superoxide is NADPH oxidase, an enzyme that generates superoxide as a part of the plant's stress response. While lower levels of superoxide are essential for signaling and defense, excessive accumulation leads to oxidative stress. To quantify superoxide production, we commonly use NBT staining, that's mean nitro blue tetrazolium assay. In this method, plant tissue are incubated with NBT, a dye that reaction with superoxide. Superoxide reduce NBT to form blue formazan precipitate, which are then visualized and quantified. The intensity of the blue coloration correlates with superoxide levels, allowing researchers to access oxidative stress severity. By monitoring superoxide level, we can evaluate the extent of reactive oxygen species-mediated damage and determine how efficiently plants activate the antioxidant defense mechanism. The hydrogen peroxide is a here reactive oxygen species involved in heavy metal stress. Unlike superoxide, hydrogen peroxide is relatively stable and can diffuse across membrane, making it a crucial signaling in plant stress responses. Hydrogen peroxide is primarily formed through the enzymatic conversion, superoxide by superoxide dismutase, SOD. The reaction follows this, in this pathway. While H2O2 plays a beneficial role in activating stress response genes, excessive accumulation leads to oxidative damage, lipid peroxidation, and protein denaturation. It makes it a critical biomarkers for evaluating oxidative stress levels in plant exposed, of course, to heavy metals. To quantify hydrogen peroxide in plant tissues, we use colorimetric and histochemical assays such as potassium iodide. starch colorimetric reaction. It means that hydrogen peroxide reacts with potassium iodide, producing a yellowish brown color that can be measured spectrophotometrically. In my lab, of course, there's some test and the method is commonly used. By measuring hydrogen peroxide level, we can assess the balance between oxidative stress and antioxidant defense mechanism in plants. Understanding this balance is essential for identifying stress-resistant plant species and improving, for example, growth or plant tolerance to heavy metal contamination. To maintain cellular homeostasis, plants rely on antioxidant defense systems, which include enzymatic antioxidant. I said about superoxide dismutase as SOD, catalase CAT, and peroxidases POD, which neutralize reactive oxygen species. The next are non-enzymatic antioxidants such as glutathione, ascorbic acid, and phytohalothins, which of course help detoxify reactive oxygen species and heavy metals, especially phytohalothins. By quantifying ROS levels and analyzing antioxidant activity, we can better understand how plants respond to heavy metal stress and develop strategies to enhance their resilience, and of course detoxification by using, by production, by synthesis, phytohalothins. As we have seen, plant response to heavy metal stress production large amounts of reactive oxygen species which can cause several oxidative damage. Three majors, of course, I said about SOD, superoxide dismutase, which converts superoxide into hydrogen peroxide and oxygen, preventing excessive ROS accumulation. Catalase break down hydrogen peroxide into water and oxygen protection cell from oxidative stress. Peroxidases catalyze the reduction of hydrogen peroxide by oxidizing various substances, helping detoxify reactive oxygen species. It's, of course, very, very easy. By measuring the activity of this enzyme, we can determine how efficiently plants are responsive to metal-induced stress. Increases enzyme activity indicate that the plant is activity is scavenging reactive oxygen species, while reduced activity, it suggests, of course, that oxidative stress may be overwhelming the plant's defense system. To quantify antioxidant enzyme activity, we use spectrophotometric assay based on specific reactions of this enzyme with the substrate. The enzyme activity is typically expressed as enzyme unit per milligram of protein, providing the measure of antioxidant capacity. Of course, we must measure the total level of protein. Antioxidant procedure is very, very easy. In many publications, there are the in detail presence. In addition to enzymatic and non-enzymatic antioxidant, plants have a special mechanism for detoxifying heavy metals. It is Phytohalatins. What are phytohalatins? They are small cysteine-rich peptides derived from glutathione. They bind to heavy metals, such as, of course, cadmium, arsenic or lead, to form a PC, heavy metal, phytohalatin metal complexes, which are then substrated into vacuoles, reducing metal toxicity in the cytoplasm. General reaction of phytohalatin metal complex formation is presented. Why are phytohalatins important? Because they prevent heavy metals ion from interaction with essential cellular components. They enhance metal sequestration into vacuoles, reducing cytoplasmic toxicity. And their production is regulated by enzyme phytohalatin synthase is important to metal exposure. How are phytohalatins quantified? The measurements is analyzed by HPLC with fluorescence detection. I don't know about other methods. We use in my lab only HPLC with FLD detectors. The steps include extraction of phytohalatins from plant tissue and, of course, derivatization with mono-bromo-biman, which enchants fluorescence. It is the only one derivatizator commonly used and commonly applied. Separation and then separation is very easy separation, of course, similar like to phytohormones, purification and then detection using only HPLC systems. The long-term experiments show that, for example, phytohalatins PC2 is the mean dimer, cis and glue is more than 3 times, 4 times, maybe 6 times. Theoretically, the dimer is from 2 to 10, but usually, maximally, we measure only phytohalatins 5 in the long-term experiments. By quantifying phytohalatins levels, we can assess a plant's ability to detoxifying heavy metals and identify species with strong metal tolerance. This information is valuable for developing crops that can thrive in contaminated environmental or be used for phytoremediation. In summary, biochemical assays provide quantitative insight into plant stress responses, particularly under heavy metal exposure. By analyzing oxidative stress markers and antioxidant defense system, by, of course, non-enzymatic and enzymatic antioxidants, we can better understand how plants cope with heavy metal toxicity. Key takeaways may include oxidative stress and reactive oxygen species generation. That means heavy metals induce excessive reactive oxygen species, such as superoxide and hydrogen peroxide. And reactive oxygen species could cause lipid peroxidation. Is measured, of course, by MDA malondialdehyde, leading to the membrane damage and increased MDA levels. Electrolyte leakage is a key physiological indicator of membrane integrity loss. And then next, of course, I said about antioxidant defense mechanism is by enzymatic antioxidant, which play crucial role in reactive oxygen detoxification, and non-enzymatic antioxidant, glutathione and ascorbic acid, which of course that we can measure by HPLC. They maintain redox balance and support enzymatic activity. And the next heavy metal detoxification by phytohalatins, which bind heavy metal and facilitate the sequestration into vacuoles and reducing toxicity of heavy metals. This detoxification mechanism determines plant tolerance levels and survival under heavy metal stress. Why are these assays important? The first, of course, they help identify stress tolerance plant species. They support breeding programs for metal resistant crop. They contribute to phytoremediation strategies using plants to clean contaminated soils. Of course, not soils, maybe of water. In addition to biochemical assays, advanced spectroscopic and chromatographic techniques play a crucial role in analyzing plant stress markers under heavy metal exposure. This method provides high precision data on metal accumulation in plant tissue, oxidative stress markers, and phytohormonal balances, which regulates plant growth and stress responses. The key techniques used in the field is, of course, the HPLC used to quantify antioxidant and other secondary metabolites. GCMS identifies volatile organic compounds and lipid peroxidation byproducts. And ICP-MS, ICP-MS, which measure heavy metal concentration with high sensitivity, is a better instrument than AS, and the Fourier transform infrared spectroscopy, which detects biochemical changes in protein, lipids, and carbohydrates. One of the most used techniques for plant stress analysis is HPLC. Why HPLC is important? It's essential for studying heavy metal response because it helps measure antioxidant compounds, including glutathione. I said about it, and phytohalatins, which help detoxify metals. How does HPLC work in these measurements? Sample extraction, plants extract containing target compounds are prepared, separation on HPLC column, typical C18. The sample passes through a column filled with stationary phase, mobile phase in methanol. Different molecules are separated based on the chemical properties and the detection and quantification. Of course, we must have fluorescent detectors, which is used for glutathione and phytohalatin analysis. When you use UV and mass spectrometry, we can detect, of course, plant hormones and oxidative stress markers. About detection of plant hormones, I said in the previous lecture, I don't remember when. Another powerful tool for studying plant responses to heavy metal stress is GSMS. This technique is particularly useful for identifying and quantifying volatile organic compounds. In the part, of course, phytohormones and lipid peroxidation byproducts. GCMS play an important role in plant stress research by detecting volatile phytohormones such as ethylene, methyl yasmonate, and abscisic acid, which regulates stress response. Analyze lipid peroxidation byproducts such as melatonin D-aldehyde and other, which indicate oxidative damage. Identify secondary metabolites such as terpenoid or alkaloid that contribute to plant defense mechanism. So the application of GCMS in heavy metal stress research can evaluate oxidative stress markers by detecting lipid peroxidation byproducts, and studying hormonal regulation by measuring stress-related volatiles, assessing plant metabolism, and identifying key biochemical changes under metal toxicity. I can use this molecular methods in my lab, so I would like to omit. Imaging and microscopy techniques provide visual insight into how heavy metal stress affect plant cell. These methods allow researchers to observe structural damage, metal localization, and oxidative stress responses at cellular and subcellular levels. The key microscopy techniques used in heavy metal stress as biomarkers research include fluorescence microscopy, confocal laser scanning microscopy, scanning electron microscopy, transmission electron microscopy, and X-ray fluorescence microscopy. The first detect reactive oxygen species accumulation and stress response using specific fluorescence dyes. The next confocal laser scanning microscopy provides a high resolution image of heavy metals distribution and antioxidative stress markers. On the other hand, scanning electron microscopy examines surface damage in levels root and stems. Transmission electron microscopy (TEMM) reveals ultrastructural changes such as chloroplast degradation and vascular sequestration of metals. And the last, X-ray fluorescence microscopy, maps the localization of heavy metals within plant tissue. Let's go to conclusion. Let's reflect on the biggest picture. How do plants survive in metal-contaminated environments, and how we can learn from them? Heavy metal stress is a silent threat to agriculture and ecosystem. Heavy metals such as cadmium, lead, mercury, arsenic don't just accumulate in soil. They invade the plant cells, disturb metabolism, and threaten food security. Yet plants have enveloped an arsenal of defense mechanisms to fight back. What have we discovered? The first, of course, the heavy metal trigger a storm of reactive oxygen species, damaging protein, DNA, and membranes. They hijack nutrients transport system, leading to defenses in calcium, zinc, and iron. They disturb, of course, heavy metals, photosynthesis, hormone balances, stunting growth and lowering yields. Antioxidant various, for example, SOD, CAT, and POD, neutralize reactive oxygen species before they cause irreversible damage. Molecular mechanism system such as glutathione, ascorbic acid, phytohalothins trap and detoxify heavy metals. Gene activation plants fine-tune their stress response in real time. And the next, of course, HPLC, GCMS methods, techniques reveal the hidden changes in stress metabolism. ICP-MS, for example, map heavy metal accumulation at the atomic level. Fluorescence and electron microscopy give us a close-up view of plant cells fighting for survival. Questions. Can we engineer crop to be more resistant to plant metals? The answer is yes. How can we use plant to clean the contaminated soils through phytoremediation? Please answer yourself. Plants have been adapting to stress for a million years now. It's our turn to learn from them. By integrating biochemical, molecular, and imaging techniques, we can develop sustainable strategies for protein crops, restoring ecosystem, and ensuring global food security. Oh, thank you very much.
Thank you, Professor, for your nice lecture. The next lecture is of Professor Qazi Fariduddin. I think he is just coming. So just 10 minutes gap. 10:25 or 10:30 he will start. Okay.
Good morning to all of you. Today I will be discussing polyamines allies in plant resilience against abiotic stress. Now I will discuss first with the last keyword of my topic, that is what is stress. So you might be aware that stress could be any external factors that disrupt the normal growth, development, functioning of the plant. Another question arises that how plant face these types of stress, what could be the source. Based on the type of source, these stresses could be biotic and abiotic stresses. So generally biotic stresses are caused by living organisms like pathogens. It includes bacteria, fungi, viruses, pests like insects, nematodes, and competition from weeds also comes under the category of biotic stresses. Abiotic stresses, as you know that this is generally induced by non-living sources. This could be induced by environmental like drought, salinity, extreme temperatures. And this could be of chemical nature like heavy metals, pollution, and nutrient imbalance. So how these biotic and abiotic stresses, they are relevant in context to plants. So you will be astonished to know that these two types of biotic and abiotic stresses can reduce plant productivity by 65 to 87% depending upon the type of crop. So how these biotic and abiotic stresses could hamper the yield of the plant. So in this way, we should dig into more details how plants are exposed to wide range of abiotic stresses, biotic and abiotic stresses that significantly affect their growth, development, and productivity. These abiotic stresses include drought, salinity, extreme temperature, heavy metals, toxicity, and nutrient deficiencies, and this disrupt the physiological and metabolic processes of the plants. Among these, drought and salinity are particularly very detrimental, reducing photosynthesis and water use efficiency. Whereas, on the other hand, biotic stresses generally results from interaction with the non-living organisms such as pathogen, insects, nematodes, and weeds. So these stresses generally compromise plant immunity, leading to very significant loss of yield. But sometimes what happens, the combined impact of biotic and abiotic stresses could be additive, synergistic, and these leads to further damage of the crop. It means that at a time plant could face both biotic and abiotic stresses. It's generally not very common. Sometimes what happens that plants only not face biotic stresses but also abiotic stresses. So what would be the repercussion of these biotic and abiotic stresses? So this could be additive, means 1 plus 1 is equal to 2. This could be synergistic, 1 plus 1 is equal to 3, and so on. So if it is the impact of biotic and abiotic stresses becomes synergistics, it's very fatal to the plant productivity. Now come to, in nutshell, I will just outline what are these, how these abiotic stresses impact the crop plants. So first I will take up salinity stress, generally disrupts the osmotic balance by reducing water availability and causing iron toxicity. So how plant responds to salinity stress? Generally, plant respond to salinity stress by synthesizing osmolytes like proline, glycine, betaine, and activate ion transporters to maintain cellular homeostasis. Then another stress is drought stress, generally reduces the availability of water, decreases photosynthesis, and generally drought stress leads to closure of stomata, rolling of their leaves in just sort of conserving water. Uh, Another stress is heat stress. Generally, once plant face heat stress, generally heat stress accelerates rate of transpiration and loss of water. Whereas transpiration is triggered, so plants generally counter these heat stress by synthesizing specific heat shock proteins and acclimate themselves by synthesizing these specific proteins. Now, plants also face chilling stress, and chilling effects stress affect fluidity of membrane, activity of enzymes, causing formation of ice crystals which damage or affect the ascent of sap, then leading to metabolic imbalance. And high light intensity generally causes photoinhibition, oxidative damage to the plants. So plants also respond to these abiotic stresses by synthesizing antioxidants and adjust arrangement of chloroplast. Whereas you see that heavy metal toxicity generally disrupts the enzyme activity and generate reactive oxygen species, triggering plants to produce metal chelators like phytochelatins and further enhance antioxidant defense system. You see that plant also face submergence stress during flooding. So what happens during submergence? Once plants are submerged, they don't get proper oxygen and face condition of hypoxia, means less oxygen for respiration. So you see that plant adapt themselves by developing parenchyma, activating aerobic metabolic pathways. So these adaptive mechanism generally adopted by plants to cope with these abiotic stresses. And the way plant overcome these stresses is very interesting to know that how plant overcome to the hostile environment. You see this, see the photograph on one side. You see that there is a drought affected wheat crop, and how severely drought affects the wheat crop is evident from this slide. And on the right side you see that tomato crop is affected by drought stress. See the devastation made by the drought stress on tomato crop. So this is just mere two examples I am showing you that how drought stress affect wheat and tomato crop. Now come to that ROS, reactive oxygen species dynamics. Generally, reactive oxygen species are very central to the plant response to abiotic stresses such as climate driven stresses like flooding, freezing, ozone, ultraviolet radiation. This could be also triggered by anthropogenic processes like drought, heavy metal, salinity, pesticide, herbicides, and soil driven stresses like salinity and change of pH. So what is the fate of production of reactive oxygen species is very crucial. So you see that under normal condition reactive oxygen species are generally produced during normal metabolic reactions as a byproduct, and these reactive oxygen species play a very significant role as a signaling molecule and control growth and development of plants. So this is the case under normal condition when ROS are produced. But what happens during abiotic stresses, excessive accumulation of reactive oxygen species disrupts cellular redox homeostasis, leading to oxidative stress that damage lipids, proteins, and nucleic acid. So in one way, once reactive oxygen species are produced under normal condition, what happens? It acts as a signaling molecule. Whereas under abiotic stress condition, when there is excessive accumulation of ROS, it damages lipids, proteins. So you see that how reactive oxygen species are produced in chloroplast. Generally, during various abiotic stresses like salinity, drought, it leads to accumulation of RS in chloroplast during photosynthesis. So what happens during excessive light generally generates singlet oxygen species and superoxide radicals in photosystem first, and second, then superoxide dismutase generally converts superoxide radicals into hydrogen peroxide, which acts both as a signaling molecule and a potential oxidant. Now, what is the fate of reactive oxygen species in mitochondria? In mitochondria, generally what happens during various abiotic stresses like salinity or temperature extremes, ROS generally form during the mitochondrial electron transport chain, and generally mitochondrial production of reactive oxygen species is very much closely linked with the metabolism and stress signaling pathway. Now you see that what happens in peroxisome during abiotic stresses like drought and highlight, photorespiration in peroxisome is generally triggered and leading to increased production of hydrogen peroxide by glycolate oxalate. Peroxisomal reactive oxygen species are very much critical for metabolic adjustment during stress. Now what happens in apoplast? So during salinity and drought and heavy metal stress activates NADPH oxidase in apoplast, and the increased activity of NADPH oxidase generally generates superoxide, which is then converted into hydrogen peroxide by superoxide dismutase. So generally accumulation of ROS in apoplast contribute to cell wall modification, and you see that stress signaling and defense responses. So other organelles like endoplasmic reticulum also produces reactive oxygen species during folding of proteins, whereas nucleus receives reactive oxygen species signals. Generally these are perceived by different genes, specific genes. So the way plant generally cope with these excessive production of reactive oxygen species. So what is the fate of these reactive oxygen species, how they are changing different metabolic pathways in the plants? So generally reactive oxygen species serves as a secondary messenger in stress signaling pathways, activating transcription factors and genes involved in stress tolerance. So this is the dual role of reactive oxygen species, which highlight the complex and vital functions in abiotic stress adaptations. Now you see that in this slide, when plants are exposed to high light stress, what is the fate of photosystem 1 and 2. So once plants are exposed to high light stress, the oxygen evolving complex in chloroplast generally absorb excess light, which damages the oxygen evolving complex and the photosystem 2 reaction center. Then it breaks down important proteins also like D1 and D2 protein, leading to photoinhibition. What is photoinhibition? That is slow down of the photosynthesis. And abiotic stresses like drought and salinity further aggravate this problem by producing the reactive oxygen species. So generally this disturbs the balance inside the chloroplast, damaging reaction centers and blocking their repairs. So this further aggravates the problem, and these stresses also harms the structure of chloroplast, causing it to shrink or swell, breaking down its layer and pigment protein complex like chlorophyll. So as a result, photosynthesis slows down, and you see that these stresses also interfere with the light reaction in photosynthesis. They block the electron transfer, stop enzymes from working and reduce production of ATP, ultimately halting the plant's ability to make food. So what is the why abiotic stress tolerance is crucial for plants? So this is a very important challenge. You will be surprised to know the data that impact of drought stress on production of certain crops. I will just share that by 2050, the global population is expected to reach 9.9 billion, creating a, you see that massive demand for food. So to meet this burgeoning population, the food production needs to increase by 60% as compared to today. And currently crops like rice, wheat, maize and soya are only increasing at the rate of 1.6% per year. So to meet the future needs, the growth rate must be at least 2.4% annually. Like major crops like rice, which feeds about more than 3.5 billion people, is growing just at 1% annually. And you see that these four staple crops provide about 2/3 of the world's calories, making their productivity is very crucial. So climate change and outdated agriculture practices are making or you can say worsening this problem more. So by 2050, we will need to double grain production from 2 billion to 4 billion tons annually to feed our population. So to overcome these challenges, how these abiotic stresses challenges could be overcome, various omics approaches are essential for studying plant response to abiotic stresses and improving agronomic traits like stress tolerance, like plant height, grain quality and yield. So genomic studies generally focuses the entire DNA of a plant to identify genes linked to abiotic stress tolerance. Various techniques like quantitative trait loci mapping, genome-wide association studies and next generation genome sequencing generally address these stress-related genes, and these tools provide further insight into traits like drought tolerance to salinity, heat and other stresses. So genomics approach also supports modern breeding methods like marker assisted selection to develop stress resilient crop. Metabolomics is very much significant, which has been magnificently applied to plants to detect novel metabolites, genes and their metabolic pathways. this is my current thrust area where I am working nowadays. So you see that metabolics approach is very crucial. All of you just pay attention to this approach because this is very crucial to address how metabolite profile changes in response to various abiotic stresses. So metabolites produced by plants which play a crucial role in stress tolerance or stress responses. By analyzing changes in metabolites like sugars, amino acids, and organic acids under stress condition, generally biochemical pathways are changes during various stress adaptation. For example, accumulation of osmolytes like proline and glycine betaine helps plant to retain water during drought stress. So metabolomics also reveal that how plants manage energy production and detoxify harmful substances under stresses. So these informations could be very much helpful for plant breeders and engineer crops with enhanced stress tolerance by targeting key metabolic pathways. So abiotic stresses significantly affect plant growth and reduces plant productivity. And so to combat these challenges, plants generally use signaling molecules such as phytohormones, and these natural regulators can mediate stress tolerance by triggering various adaptive responses. Phytohormones like abscisic acid, auxins, gibberellins help plants to adjust to these abiotic stresses. For example, ABA helps to reduce water loss during drought stress by closing stomata, whereas auxin and cytokinin regulate growth and stress-induced expression of genes. So by leveraging phytohormones, we can develop strategies to improve plant resilience and enhance crop production under stressful condition. So phytohormones are basically very much essential regulators that help plant to adapt and survive under abiotic stress condition by activating various protective mechanisms. So out of these phytohormones that are employed to cope these challenges, I will only confine to polyamines. So polyamines, they are different amino group like putrescine having is a diamine, spermidine, spermine. These play a very crucial role in regulating plant growth and various stress responses. So polyamines basically protect plants from oxidative damage, stabilize cell membranes, and promote root growth. So these polyamines also improve photosynthetic efficiency by protecting chloroplast by enhancing pigment synthesis. So you see that polyamines, melatonin, brassinosteroids, salicylic acid, say these they work together to promote key physiological processes in plants. So cumulatively, their effect could be additive, synergistic, and all these molecules like melatonin, brassinosteroids, salicylic acid, and polyamines, they improve gas exchange parameters, enhance pigment content, and regulate root architecture for better water and nutrient uptake, and develop enzymatic system of the plant to cope with these challenges. Additionally, these molecules also help in reducing heavy metal uptake, supporting the survival of the plant under challenging environmental conditions. So I am going to discuss in more detail regarding the role of polyamines in plants. So just you see that these polyamines, they are found in living organisms, both in plants and animals, and even in some viruses. In plants, generally these polyamines, they are present in free form, and the most common polyamines in plants are putrescine, spermine and cadaverine. Uh, but you see that these polyamines, they are distributed differentially across the plant tissues, organs, and at different growth stages. For example, you see in tobacco, uh, putrescine and spermine are more abundant in young leaves and shoot tips, whereas spermidine is higher in older leaves. So higher polyamines like spermidine and spermine are mostly produced in aerial parts of the plant like shoot and apical meristem, whereas putrescine is mainly produced in underground parts such as roots. So among these main polyamines, uh, generally spermine and spermidine are more evenly spread throughout the plants than putrescine. Uh, Polyamines generally also accumulate in different parts of the plant cells, like in carrot cells. For example, putrescine is mainly found in cytoplasm, whereas spermidine in the cell wall. So you see how there is a differential accumulation of polyamines in plants. Now, this is the image retracted from one of my recently published paper in chemosphere. So you see that here polyamines biosynthesis generally you see that begins with the decarboxylation of amino acid ornithine or arginine, and thereby producing putrescine is a simple diamine. So you see in this figure that this process could happen through two pathways. One is known as ADC pathway and another one is known as ODC pathway. So polyamines could be synthesized by adapting these two basic pathway. One is ADC pathway, another one is ODC pathway. So several enzymes work together to produce putrescine. After that, spermidine and spermine are created by adding aminopropyl groups to putrescine with the help of enzyme like spermidine synthase and spermine synthase. So the aminopropyl groups comes from methionine, which is processed by SAM decarboxylase. Under these conditions, what happens? Plant regulate generally level of polyamine through catabolic pathways. So you see that biosynthesis and catabolic pathway run in a balanced condition to maintain proper homeostasis of polyamines. So these pathways generally involves enzymes such as DO and PO, which breaks down the polyamine and produces byproducts like pyrroline, ammonia and hydrogen peroxide. So in catabolism of polyamines, you see that one of the reactive oxygen species, one of the antioxidant is produced, namely hydrogen peroxide. So you see that the accumulation of hydrogen peroxide can cause damage to various lipids, membranes, and it also plays very important role in strengthening the cell wall of the plants, which helps to confer tolerance to the plants. So the level of polyamine in plant cells are generally very much tightly regulated through their synthesis or breakdown, ensuring that the plants has the right balance of polyamines. So this balance is basically very much essential for plant growth and ability to cope with the challenging environment. And generally polyamine homeostasis is maintained by a complex feedback systems that control the production, breakdown and movement in and out of the cells. So in addition to this, polyamines are also involved in producing their other biologically active compounds and support plant health and confer stress tolerance. So you see that polyamines, they are involved in regulating various physiological processes of the plants like growth and development, where they regulate cell division, elongation, differentiation, support the root and shoot growth, flower development and fruit ripening. And in addition to this, polyamines also regulate various abiotic stress responses like just I mentioned drought, salinity, and these polyamines basically stabilize cell membranes, scavenge reactive oxygen species, and enhance antioxidant defense activity to protect plant from damage. So these polyamines could be also involved in mediating defense responses like polyamines contribute to plant immunity by strengthening the cell walls through lignification, triggering defense related signaling pathways and producing reactive oxygen species to combat pathogen attack. So how these polyamines help in to delay senescence? So generally polyamines delay senescence and aging by reducing oxidative damage, by maintaining proper chlorophyll content and protecting cellular homeostasis for further adaptability to the plants. Now this slide shows the impact of polyamines in different plant species grown under heavy metal stress. Various case studies have been cited in this table regarding the role of polyamines in heavy metal stresses like cadmium, chromium, lead and aluminum on different crops like wheat, rice and vigna. So just to have an overview of how polyamines modulate different abiotic stress in plant cell in a nutshell, you can just have an overview through this table. Now, role of polyamines in further in stress tolerance, how polyamines mitigate various stresses like salinity, water and heavy metal stress. They are further summarized in this table. Now just I will take a few case studies done from my lab. So what is the role of polyamines like putrescine and spermidine in mitigating manganese induced toxicity in Brassica junctia? So that I will discuss. So what happens once plant faces toxicity of manganese, what happens? It leads to the development of brown speckles. You see on the brown speckles here. And generally this brown speckles develop on mature leaves. So this is the case with manganese toxicity. So the intensity of brown speckles formation generally could be used as a simple and rapid method for evaluating various cultivar for manganese tolerance. In addition to this, intraveinal chlorosis and necrosis are additional symptoms of manganese toxicity, and these symptoms may be accompanied by distortion in the young leaves known as crinkle leaf. So this crinkle leaf is one of the repercussion of manganese toxicity, which is characteristic indication of calcium deficiency also. And this manganese toxicity generally often leads to deficiency of other nutrients like calcium, magnesium, iron and zinc. So generally the induced deficiency of iron and magnesium results from inhibited uptake across the plasma membrane and competition or imbalance at different cellular levels. Additionally, this manganese toxicity can also lead to loss of apical dominance and increased formation of auxiliary shoots, and this condition is referred as witch's broom. You see in this figure witch's broom. So in this slide you see that how manganese toxicity is affected in different crop plants, how photosynthesis is modulated. So just quick overview of manganese toxicity in different crop plants could be generally retracted from this table. So you see, this is the finding from my lab that how the polyamines modulate uh, response of plant, particularly various growth traits like length and root of shoot, uh, length of root and shoot, fresh and dry mass of the root and shoot, and leaf area. And you see that all these parameters decrease in presence of higher concentration of manganese, but how application of spermidine at 1 millimole just recovered the damage induced by higher concentration of manganese. The other parameters like leaf area, SPAD value and photosynthetic traits, they were also considerably reduced at higher concentration of manganese, and these stress plants once treated with spermidine or putrescine, these counteracted the toxicity induced by higher concentration of manganese. So you see, this slide shows the impact of stomatal behavior under higher concentration of manganese. Generally, what happens at higher dose of manganese leads to closure of stomata, whereas you see that later on exogenous application of putrescine, spermidine or putrescine generally restored the damage induced by higher concentration of manganese. Now, this slide shows the application of spermidine or putrescine alone in stress-free plants, which generally enhance the photosynthetic efficiency of PS2 as compared to control. And generally, under unstressed conditions, spermidine and putrescine generally increased these parameters, and once spermidine and putrescine were applied on the stress plants that have already been given higher concentration of manganese that overcome the toxicity generated by manganese. Then come to the antioxidant enzymes. Antioxidant enzymes were further triggered in response to higher dose of manganese, and generally this application of putrescine or spermidine generated additive effect and on the antioxidant activity. So this is the model showing the how polyamines plant responds to polyamine and heavy metal stress. Generally, plants exposed to heavy metal stress changes the level of phytohormones, generates production of reactive oxygen species leading to oxidative stress, and further activation of defense system are triggered generally. Heavy metal stress generally affect the metabolism of polyamines, affecting both synthesis and its breakdown, that is catabolism. The activity of various enzymes like ADC and other enzymes generally increased, leading to an increased production of putrescine levels, and putrescine generally acts as a signal indicating the presence of stress, which in turn enhances the biosynthesis of other polyamines like spermidine and spermine. Basically, spermidine and spermine are primarily responsible for regulating stress responses, interacting with reactive oxygen species, phytohormones and transcription factors, and other molecules to manage the both stress responses and various essential cellular processes. Metal stress also increase polyamine catabolism through increased level of enzyme activity, producing beneficial byproducts like GABA and hydrogen peroxide. High polyamine levels generally activated oxidative system defense mechanism through production of hydrogen peroxide mediated signaling, scavenging of reactive oxygen species, interaction with other enzymes, and protection of cellular components from oxidative damage. So there is also proposed role of polyamines directly in heavy metal stress, although further research is needed to confirm this hypothesis. And this polyamines also enhance the glutathione levels, boosting the biosynthesis of phytogelatin conjugates, which generally activate heavy metal detoxification. So, what is the conclusion of this finding, that how manganese stress affect the plant? That manganese stress reduce the movement of stomata, decreased photosynthetic traits, lowered the biochemical parameters in Brassica junctia. And then what is the impact of polyamines, spermidine or putrescine application? It led to the improved stomatal movement, photosynthesis and growth rates were enhanced significantly, and antioxidant systems were improved, reducing oxidative damage. And then one significant aspect is application of polyamines decrease the accumulation of heavy metals. So if I talk about the comparative effectiveness of two polyamines, I have taken spermidine or putrescine. So out of these two polyamines, spermidine were found more effective than putrescine in mitigating manganese-induced stress and restoring plant health. So this is the role of brassinosteroid and spermidine in combination in mitigating manganese stress in Brassica junctia. I will skip this. This is due to time constraint. Now, what is the role of polyamines in manganese-induced toxicity? So, you see that higher concentration of manganese supplied to Brassica junctia through soil generally reduce the root morphology, reduce the growth, distorted the root morphology and altered the stomatal behavior. And application of spermidine through foliage proved very beneficial and reduced the production of reactive oxygen species, and this was further confirmed by the microscopic studies. What was the fate of stomatal movement? Now, advanced molecular approaches such as transgenics, proteomics, metabolomics, they are also being employed to manipulate metabolism of polyamines and to study their role in stress tolerance. Now, next question arises, how polyamines metabolism could be manipulated for conferring abiotic stress tolerance. I am only focusing polyamines, how its metabolism could be manipulated. So generally, various studies have shown that particularly in Arabidopsis, that the production of polyamine is affected by different environmental stresses. So it was revealed that certain polyamine producing genes, they are turned off and they struggle more with the stress, suggesting that polyamines play an important role in helping plants to resist various sort of stresses. Additionally, genes responsible for production of polyamines from various organisms, they are further incorporated into plants, and then plants tend to show better stress tolerance and resistance. This is linked to the level of polyamines. So if I ask you what is the role of polyamines level in plants, so polyamines level is very important in plants in mitigating various abiotic stresses by regulating both its biosynthesis as well as catabolism. So, in addition to this, various stress related metabolites, they are upregulated by application of exogenous polyamines. They regulate different metabolites that enhance photosynthesis in plants like in cucumber. Also in other stresses like salt stress by regulating stomatal opening and closing. Polyamines also regulate different metabolites that help in the expression of polyamines like diamine oxidase, polyamine oxidase, NADPH oxidase, and also modulate the production of hydrogen peroxide. So, exogenous polyamines generally help plants to tolerate metal stress by regulating different metabolites and thereby protecting cell membranes by forming various complexes like phospholipids and with other metals. They boost glyoxylate system activating enzymes like GLY1 and GLY2 to detoxify harmful methyl glyoxal. So, in this way polyamines also increase the level of other metabolites such as glutathione, which help in metallocylation and transport of detoxification. They improve the activity of various enzymes just I mentioned previously like SOD, catalase, ascorbate peroxidase, and non-enzymatic antioxidant like ascorbate reducing oxidative stress. So I will skip this. So you see this table shows the list of metabolites differentially accumulated in tomato leaves and how various polyamines alter the metabolic profile in tomato plants. So you see that through these whatever the metabolite is expressed in higher concentration that could be sorted, and then a specific pathway could be drawn that how these pathways are altered and which intermediate is highly expressed, and accordingly the enzymes could be identified so that the problem could be correlated that how polyamines altered the various metabolic pathways in the plants. So by identification of wide range of differentially expressed proteins and their further exploration of molecular mechanism also reveal more detail about how polyamines regulate various metabolic pathways. Now I would conclude that high polyamine content for short-term stress is beneficial for the plant, and elevated polyamines level either through exogenous supply or through genetic manipulation confer tolerance to short-term exposure to the plants grown under various abiotic stress conditions. In addition to this, application of spermidine or putrescine or both to stressed and stress-free plants modified the movement of stomata, improved photosynthesis and growth rates, and also enhanced antioxidant system, whereas accumulation of manganese and production of superoxide radicals were reduced. And out of, if I consider out of these two polyamines which I had discussed like spermidine or putrescine, spermidine was found more significant, prominent in restoring the damage caused by manganese. So what are the future challenges? That unraveling various metabolic pathways with regulatory nodes by adopting metabolic approaches will going to help to identify unique or shared regulatory nodes within metabolic pathways affected by polyamine metabolism, which will further decipher details and crosstalk among different pathways that influence various abiotic stresses. Then, in addition to this, targeted genetic engineering for conferring stress tolerance is further needs to be addressed by identifying the key regulatory targets in metabolism of polyamines will further reveal the development of genetically engineered plants to confer tolerance under abiotic stresses. Then there is a need to optimize regulation of polyamines for various stress responses. So this could be the regulation polyamines level in the plants could be controlled by controlling various the specific metabolism related to polyamines in a controlled manner or tissue specific manner by activated early response in stress tolerance. So this is going to ensure plants to produce polyamines in controlled way as desired without disrupting normal growth conditions or metabolic pathways. Thank you. Exactly 1 hour, I think. Not 1 hour. Hmm? इसको 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 दे रखो. So now you have a tea break up to 12 noon. So you are requested to assemble here exactly at 12 noon. Okay. more details about the carrikins and some other plant growth promoters in relation to yield and quality of medicinal and aromatic plants. So I welcome you, sir, today for this informative lecture, and students try to gain what Professor Masoor Sahab is going to discuss, and try to imbibe the novel things he is going to reveal, and definitely you could incorporate these novel aspects in your future research. So, sir, you are welcome to deliver your lecture. Thank you.
ये चलेगा अच्छा स्लाइड चेंज इससे होगी ठीक Assalamu alaikum and good afternoon to everybody. So today I am going to talk on carrikins. They are also called as carrikinolites. Both are synonyms. Both are either you can call carrikins or carrikinolites. This is relatively new group of plant hormones. Okay. So basically, in Australia, there was a very much frequent phenomena of fire forest. As recently in California, there was a big calamity and huge properties were lost by the fire forest. So similarly, in Australia, that was a common phenomena. In the jungles of Australia, the fire forest was a common phenomena. So the scientists examined that after fire forest, in the next season, the growth of plants was very much abundant. So they found out that by charring and burning of leaves and twigs and wood of the jungles, some organic compounds were released by this smoke, and these compounds were very much effective in increasing the growth and particularly the seed germination. So this work was first started by some scientists of Australia. So later on, much work was done on carrikinolites, and now it has been established perfectly that carrikins, which are found in smoke water or smoke, these compounds are very much influential in enhancing the growth of plants and particularly seed germination. So by stimulating with this research, I performed this work and I designed an equipment for generation of smoke water myself. So as I have told you that carrikins were discovered by some Australian scientists, and they found out that after the fire frost, when the rainy season come, after rainy season there is huge growth of plants because those smoke compounds were retained in the soil, and soil is also a bank of seeds, a good source of seeds. So those carrikins stimulated the seed germination, and the growth of the plant was very much luxurious, and abundant growth occurred due to the action of the compound which is found in the smoke of burning of plants or leaves. So this is Australia and New Zealand. So in both places, this phenomena happened, particularly in the Western Australia. Western Australia, this invention was made. This is the Western Australia, this one. Perth is here, Perth, and this all. So the scientists of University of Western Australia, they did this research and found out that smoke of plant can stimulate the growth and germination of seed. Later on, it was found out that new group of compounds, kerikins, they are very much effective in causing the seed germination and general growth of the plants. So this word kerikin comes from Australian language, which means smoke. In fact, so this is a slide of fire forest in Australia, and most of them are eucalyptus trees. And Australia is very much famous for eucalyptus trees. You will be surprised to know that in Australia there are 900 species of eucalyptus. Right? I have also seen, I have visited Australia 2 or 3 times and found out a variety of eucalyptus. Okay? Not only tall plant, but sometimes the plants are like people and bargad and expanding, not very tall. So Australia is very much famous for eucalyptus. So by the burning of the leaves and twigs and wood, these kerikins were produced. So kerikin role in post-fire vegetation regeneration. Slide A is the blaze of blaze produces smoke and ash with kerikin. This slide here is by the blaze and the smoke is produced. Number 2 slide shows after the fire kerikins appear on the earth surface. After rainfall, the kerikins which were dissolved in the air, they come to the ground on the earth, okay, and they are stored in the ground like seeds. In the 3rd slide, following the first rain, kerikins increase soil seed bank, germination and new plant growth. You see, the 3rd slide shows the luxurious growth of new plants and high rate of seed germination occurred. So this was happened, and what led to the discovery of kerikins? For many years, it was recognized that the chemicals that a fire produced, rather than the heat, encourage the germination of some fire flower seeds. Both smoke and water that was drenched in chair or that smoke was passed through creating smoke water may have the same effect as fire. So to find the active ingredient, several teams of scientists worldwide separated the hundreds of compounds in smoke water into distinct fractions using liquid chromatography and then tested each fraction for seed germination activity. The active fractions underwent additional fractions and analysis. This investigation ultimately isolated an active molecule and chemical synthesis verified this finding by Filamenti et al. This compound contains a specific type of lactone known as butanolide fused to a pyran ring with the systematic name 2-methyl-2-hydrofluoro-2,3-pyran-2-one. Subsequently, several closely related compounds were discovered in the smoke and collectively called as kerikins. These are called as kerikins or kerikinolite. Both are synonyms. So, how this word was used, I will come to this slide, that slide later on. So, in fact, kerikin in Aborigines language is called to smoke. Smoke को ये लोग kerikin कहते थे. In fact, in Australia, the original natives were Aborigines people. Aborigines, okay. When the European people went over there, they genocided the original natives of Australia, which are Aborigines, and they killed them. And you will be surprised to know the government used to issue the license to kill the Aborigines. They have only sword or arrows, but these Europeans, they have guns, so they killed them, and many of them vanished to it. I have also seen Aborigines over there. So, so in Aborigines language, kerikin means smoke. So this kerikin word was derived from Aborigines language, which Aborigines people used to say. So therefore, these compounds are called as kerikins because they are derived from kerikins, from smoke, and the Aborigines people called the smoke as kerikins, right? So this is a slide of Aborigines people, the natives of Australia, and still there are some tribes are there. They are surviving there, but they live far from the uh, population, and they live in the jungles. So these people called as carrick to smokes. Therefore, the name of compound is kerikins. So there are many type of kerikins. KR1, KR2, KR3, KR4. Many compounds are they with little difference. For example, KR1. Okay, this is the most important kericrolide or kerikin, which is commonly found in plant smoke. KR1. KR2 is same, but one methyl group is missing over here. KR3 is the same, but one additional methyl group is attached over here. KR4 is same, but here the additional methyl group is attached over here. Similarly, these kericrolide can also be obtained by the burning of sugar. If you burn the xylose sugar, you can also obtain kerikin 1 and also estragol. So estragolectrons and kerikins are very close to in their function, and their estragolectron is also a good plant hormone. So the kerikin family, the first kerikin discovered was KR1, also known as kericrolide. Since kerikins can be produced by burning sugars such as xylose, the pyron ring of kerikins is probably derived from such pyranose sugar. Both kerikins and estragolectrons hormones such as estragol have a butylide ring. So these are the people of Aborigines. Kerikins are produced by the fire. It was discovered that the burning of many various plant products, including straw, cellulose, filter paper, and even sugars, could produce kerikins. Their synthesis from polysaccharides and sugars explain the pyron ring of kerikins, which is thought to be formed directly from pyranose sugars found in plant material. Kerikins are most likely produced at home by toasting and roasting specific plant foods. Cigarette smoke enhances seed germination, most likely because it contains kerikins. Further research has revealed that kerikins become unstable at extremely high temperatures. So this pyron ring is a common feature in the kerikins, the hexocyclic compound, and having oxygen in its heterocyclic ring. Kerikins also produced by some factories, for example, sugar factories. In sugar factories, the bagasse, उसका जो खोजड़ होता है, यानी extract निकालने के बाद में, that is known as bagasse. So bagasse is used as a fuel. Okay, when these factories are started, first they use some firewood, but later on, when this bagasse is produced in huge quantity, then these bagasse is dried in sun and used as a fuel. So by burning out these bagasse, kerikins are also produced in the environment. And after rain, the kerikins come to the soil, which can increase germination of the existing seeds. So, so I also worked on kerikin, and I designed a apparatus which can produce the smoke water which contains a lot of kerikins. So I worked on Daucoscarota, and then Trigonella funiculum, that is methi, then Corynus sativum, dhania, and then Anthem gradans, soya, and fennel. So we worked on 5 plants, and purpose of study was because lack of arable land, the arable land is insufficient. Secondly, increasing the demand of global market of the plant. Thirdly, that to increase the turnover. And fourthly, genetic potential of the plants was realized on the maximum by the fertilizers. So we can't increase the productivity by application of fertilizer because maximum genetic potential has been realized. So, to overcome these problems, we worked on this smoke water plant, and this smoke water and kerosene can break the dormancy, stimulate the seed germination, enhance the growth and productivity, help in uptake of nutrient following somatic embryogenesis, displays activity at a very low concentration, interactions with endogenous plant growth regulators, no mutagenic or genotoxic effects are reported. So there are 71 compounds reported till date, out of which kerikins and cyanohydrin are the most important, which can stimulate the seed germination. So I have told you that there are different types of kerikins, KR1, KR2, KR3, KR4, KR5, and KR6. So I have done on KR1. So this chemical was purchased by a Canadian company, Toronto Chemical Company, and you will be surprised to know that 1 milligram cost of this KR1 was 25,000 rupees, more expensive than gold. So I purchased 1 milligram from Alberta Chemical Company, and then for the sake of comparison, because if you don't have a standard, how can you compare? So therefore, we were compelled to purchase this 1 milligram of KR1 from Canada, and then we worked on smoke plants. Okay, so this apparatus was prepared by myself, and in this there is a hot plate which was purchased from a Mumbai company, and then hot plate, on the top of hot plate there is a petri dish which contains the straw, because straw, burning of straw produces kerikins. Okay, so we took the straw, clean it, and then dried it. After drying, it was put in the petri dish, and then the hot plate was turned on. By the heat, the straw was charred, was burnt into smoke, and this smoke was passed through this tube. Here is a suction flask. Okay, in suction flask we took the distilled water, DDW. Okay, and then the outlet of the suction flask was connected to a motor. I purchased this pump from USA. That was not very expensive in those days. So I used this pump, suction pump, and then the suction pump was turned on. So this smoke path was like this. It was generated by the straw, then went to this tube, and from this tube it was dissolved in water. By the suction of the air, there was bubbling in the flask. Okay? By the huge bubbling, the smoke was dissolved in water, the DW, right? But a point came when there was no dissolution of smoke, so saturation point came, and this water was called as SSW, smoke saturated water. So this smoke saturated water, SSW, was used in our study, okay, right? So after each 5 minutes, we also read the optical density of this smoke water, okay? And after about half an hour, the optical density remained constant, and this showed that no more smoke is going to be dissolved in water, and now the water is fully saturated with smoke. So this is called as SSW, and then SSW, smoke saturated water was diluted to 1000 times, 500 times, 100 times, and 250 times. And this diluted smoke saturated water was sprayed on different plants, okay, which stimulated the growth and yield of the plants, right? Shari sir, can you run this small video? I have also a small video of this. So you can see that the hot plate is working. Temperature goes to 500 to 600 degrees centigrade, and at this temperature the charring and burning of the straw starts. Smoke is produced, and this smoke passes through the distilled water because the air is sucked by the suction pump. Okay? We can regulate the speed of suction by a regulator, which is, I will show you. By this regulator, we can control the speed of supply of smoke. This is the regulator. By this regulator, we can control the speed of bubbling or the speed of smoke flow in the water. Okay? And as I have told you, that after 5 minutes, each 5 minutes, we just took a micropipette, take a sample of 0.5 ml and read the optical density using a spectrophotometer, right? So optical density was increasing, and after 35 minutes there was no increase in the optical density. It showed clearly that no smoke is dissolving in water, that is, water is fully saturated with smoke, right? Then the machine was stopped and the smoke water was taken and it was diluted to 500 times, 1 to 500, 1 to 100 times, 1000 times and 250 times, right? So we found out that dilution factor is 1:500 was most effective in obtaining the maximum seed germination and in obtaining maximum growth and yield of the plant. Okay? Okay, now we come back again to the PPT. So the whole apparatus was prepared by me, and this paper was published in Journal of Plant Growth Regulation in those days. I think about 6-7 years ago. So this is the smoke saturated water, which was filtered by Wattman filter paper. Yes, this is the main graph. You see, about 32 minutes, the water was fully saturated with smoke. So this water is called as SSW, smoke saturated water, which was used for our study, and this water was diluted to different dilutions and was sprayed on plants. Okay, so we found out that with the dilution factor 1:500 was the most effective in increasing the growth and yield of plants. Even the active constituent was also maximum at this concentration. Okay. So this work was a part of PhD program supervised by me to one of my past students, Ms. Arshia Aqil. Arshia is currently pursuing her postdoctoral research program in Copenhagen, Denmark. Nowadays she is in Denmark and doing some molecular work in Denmark as a postdoctoral fellow. So we separated this smoke water by Wattman filter paper, and then HPLC was run. HPLC was run, and we found different peaks of compounds, and then we are also very much thankful to Professor Javed Iqbal Sahab in our chemistry department. Now he is no more with us, but he was a very dedicated person. He spared days and nights to recognize that which compound is KR1. Fortunately, by using a compound library, he was successful that this compound 153 peak number was the KAR1. Okay, so in this way he was successful to identify that this peak number 153 was the peak of KAR1, which is very small amount. KR1 is found in the smoke water. So in this way, we use different dilutions of smoke water. You can see 1:1000 and 1:500, 1:250 and 1:100. And this is the control, that is the double distilled water. So similarly, we also sprayed the Carrican compound which we were purchased from Canada, Alberta University, Alberta Chemical Company, and 4 concentrations we took: K1, that is 10⁻⁻10, K2 10⁻⁹, 10 K3, 10 power 8, K4, 10 power 7. So 5 concentrations, including one control, we took from the smoke water, and 5 concentrations we took from the kerrikins which we purchased from Canada. Okay, one is control, and 4 concentrations were taken. So we found out that 10 to the power minus 8 concentration, and in some cases 10 to the power minus 9 concentration of kerrikin, and in case of smoke water 1 is to 500 in most of the cases, and occasionally in 1 is to 250 ratio of smoke water proved best for the increasing the growth, yield and quality of plant, and also the seed germination. So we performed many experiments on different plants, and in 5 plants we took 2 sets, 1 set for smoke water and 1 set for kerrikins, right? Smoke water, kerrikin, smoke water, kerrikin. In this way 10 experiments were performed on 5 plants. Clear? So we performed 10 experiments, and for each plant we took 2 sets of experiments. One set was designed for smoke saturated water, dilute factors, and one set was carried out for the kerrikins as in for the sake of comparison. So in a nutshell, we found out that smoke water was most effective in comparison to kerrikin alone was applied to the plant, because smoke water contains so many kerrikins. Therefore, obviously, its effect was most obvious and clear in comparison to KR1. But as far as the control is concerned, KR1 10 power minus 8 or 10 power minus 9 gave the maximum performance of the plant, including seed germination also affected. We studied many parameters of these plants, many photosynthetic parameters and phenol content, DPPH activity, and yield parameters, and seed germination parameter, etc. So in this way, we found out that out of SSW, 1 is to 500 dilution gave the maximum germination, you can see, right? In case of kerrikin, 10 power minus 8 gave the maximum germination value in case of 10 power minus 8 molar concentration of kerrikin. Similarly, chlorophyll content and other parameters, they gave the highest value at 1 is to 500 in case of smoke saturated water, and in case of kerrikin, 10 power minus 9 or 10 power minus 8 gave the maximum value. Plant weight, root length, diameter, fresh weight, root fresh weight. Similarly, for photosynthetic parameters also gave the maximum value in case of 10 power minus 9 minus 8 molar, and in case of smoke water 1 is to 500 dilution was the best. So different parameters were found best by the application of smoke water as well as the kerrikin. So the trigonella, that is methi, contains a compound known as trigoline. Trigoline content was also increased by the application of kerrikins and smoke water. Okay, for example, here 2.6 milligram in control and 3.17 milligram per gram in case of they are treated by KR1 by smoke water. Similarly, by the application of KR1, it was 2.6 in control and 3.1 milligram in case of kerrikin. So how does kerrikin works? This is still a question, but many of the steps have been discovered, and according to this, this is the action of the kerrikin and this is the action of strigolactones. Because both actions are more or less similar, therefore both are studied here. So this is the application of kerrikin, that is the application of strigolactones. In this, this step is still not discovered that what what precursors are involved in the formation of these receptors, in which one is ubiquinone is very much common to both of the compounds. In this way, both the compounds they produce some secondary messengers. Okay, in case of kerikins, it is like this. In case of stereoelectrons, it is like this. These secondary messengers go to the DNA and then they stimulate the DNA replication, and in this way the proteins are produced. Those proteins either they stimulate seed germination, or they induce the root system architecture, or they induce the mycorrhizal symbiosis. So in this way, this is the mechanism how kerikins stimulate the seed germination and growth of the plants. So now, as my title of my talk is not only confined to kerikins, I want to tell you that we have also worked on some other plant growth promoters, and now it comes a new technique of promoting plants by RPPs. RPPs mean radiation processed polysaccharides. Okay, so this is another group on which we worked about 2 decades, and we were granted 2 major projects by the Biochemical Center. And 2006 they gave the first project on mint using RPPs, and the results were very encouraging. Then they said, now you should perform the experiment in the field. Okay, the first project was concerned with the pot experiment. When the results were encouraging, they asked us to do the field. Then we went to Sambhal, because Sambhal is a land of mint, because there in the agroclimate is very much suitable. Okay, therefore the BRC people asked that you should perform this experiment in that area where the mentha is grown very luxuriously and successfully. Therefore, we went to Sambhal. We land a piece of land of 7 acres, and we performed field experiment over there. So basically, there are some polysaccharides, particularly marine polysaccharides like carrageenan, alginate. These are found in seaweeds. Okay, seaweeds produce lot of polysaccharides like sodium alginate, carrageenan, okay, and so many. So when these separated or extracted polysaccharides are subjected to gamma radiations, these polysaccharides are broken down into smaller molecules. ठीक है ना, जैसे आप किसी शॉपकीपर के पास जाएं कि भाई ज़रा 1000 का चेंज दे दो, तो वो 500 का, 100 का, 200 का नोट दे देगा आपको. Similarly, by the action of gamma rays, the large molecules are broken down, they are decomposed to smaller molecules. These water-soluble smaller molecules, when applied to the plant, they can increase the growth and yield of the plant. Okay? So initially this work was done in some like Manila and Philippine countries. So then I submitted a project to BRC that if we apply this technique to medicinal plant, can we increase their yield and quality? So they appreciated this idea very much, and they gave this project. So we worked on sodium alginate, carrageenan, and chitosan. Chitosan is also a polysaccharide, but this is an animal-derived polysaccharide, which comes from crabs and shrimps. Okay? समंदर किनारे जब waves आती हैं, तो they bring the small bodies of these shrimps and prawns and crabs. So when the waves go back, the laborers pick those dead bodies of shrimps and prawn, and they collect in the sacks. Okay, and then these are taken to the factories. In factories, they are broken down into small pieces and then boiled. Okay, in the boiler. After boiling, the chitosan is extracted. Okay, now this chitosan is purchased by Sigma companies like that, and then we purchase it from Sigma company. The chitosan then we send to Biochemical Center. In Biochemical Center, they expose the chitosan to the low dose of gamma radiation, okay, about 200 kilogray or so. In this way, the large molecules were broken down, depolymerized to smaller molecules, right? So when these smaller molecules are dissolved in water and sprayed on the plant, they increase the growth, yield, and quality of the plant. Okay? Alginate and carrageenan are water soluble, but however, chitosan is not water soluble. You have to use some acetic acid to dissolve it into water. Okay? So we dissolve this radiation process polysaccharide into 0.3% acetic acid solution, and then it was successfully dissolved. Then we sprayed these solution on the plant, and we found very useful and very spectacular results of these polysaccharides, right? You got the point? So we did not use radiation on the plant directly. We realized the benefit of radiation indirectly, okay? We did not expose the plant to the radiation, rather we use radiation technique to only break down the larger molecules into minute molecules, right? So in this way, there was no effect of radiation on the plants, or the product of this plant will not harm any human being which is going to consume it, right? So this is totally safe from radiation effect. We use radiation only to break the big molecules into smaller ones, right? So we use this work on mentha, and mentha contains a lot of menthol, which is used to prepare balms and wicks and many cough syrups, okay, which is very much effective in making cough syrup, like, okay. So we have collaboration with BARC, and Dr. Lalit Varshini was my principal collaborator, and he was a good scientist. His base was chemistry, and then we worked on this project, and we found very interesting results on radiation process polysaccharides. So in this way, we worked. So I would like to tell you that these large molecules can also be broken down by different methods. For example, you can use the bacteria, or you can use other technique, but this technique by breaking the molecules by radiation is very much safe and quick and very successful. Okay, you can break these polysaccharides by gamma rays or electron beams. Okay, and in this way we can found these smaller molecules which show antimicrobial activity, which also show illicit activity in plant, promotion of growth, suppression of heavy metal stress. So these compounds are very much useful in solving many agricultural issues. So first is sodium alginate. So we also did the GPC of this compound, and then is chitosan. As I told you, that this is animal product, chitosan, but you should know that effect of chitosan on plants was very much overwhelming than the carrageenan and sodium alginate, right? So it was dissolved in acetic acid, and then it was sprayed on the plant. So different plants were chalked out, and then we sprayed these chemicals on plant, and then we were successful in increasing different parameters like growth parameters, biochemical parameters, yield parameters were increased. This is the box of sodium alginate, and this is the sodium alginate in a petri dish. So these molecules are very large in size. You see, only one unit has been shown, but thousands of units are there. By the action of radiation, these long chains are broken down to smaller molecules, which are known as radiation process polysaccharides, or you can also call them oligosaccharides. Oligosaccharides mean smaller broken molecules. So these smaller broken molecules were dissolved in water or acetic acid and then sprayed on the plant. This is gamma rated in BRC, where these chemicals were broken down into smaller molecules. Okay, these are the images of electronograph before breaking and after breaking, right? So then these chemicals were dissolved either in water or acetic acid and then sprayed on the plant. So this project we perform at Sambal, about 200 kilometers away from here in the north direction, and this area is very much famous for mint production, right? And then this was a project run on the mint. This is my student Hassan Jalil from Jammu, okay, in this project. Then the chemical was sprayed by using electric spray machine. The machine was charged at night, and the daytime the sprays were made on the plant. You can see very huge and very luxuriant growth of the plants were happened. The nearby farmers were also surprised to see that, sir, your plants are very much healthy than our plant, and moreover in your plant there is no insect attack, but our plants are also affected by insects. Okay, so we can obtain two benefits. Number one, high growth, and these chemicals were also used as pesticides. Okay, so both the benefits were obtained. This was project on mint. You see control plant gave this much oil, and then 40 ppm gave this much oil, and this 60 ppm gave more oil, and 80 ppm gave the maximum oil production in case of chitosan application. So then the oil was run for GCMS, and the fractions were separated and quantified by GCMS machine. This GCMS is also sanctioned by the BRC project. We purchased so many equipment by this project, GCMS, spectrophotometer, and so many smaller equipment. So in this way, we perform this experiment in the Sambal in 3 types of plots. Okay, 10 square meters, 100 square meters, and then 1000 square meters. Because bar people said that you should do this experiment in the field, and you should increase the site so that we can confirm that the results are reliable and valid. So therefore, for 100 square meter plot, we purchased this equipment from CIMAB Lucknow, and in this way, yes. So this distillation plant was for 15 kg of biomass. You can put 15 kg leaves of mint in this container, and this was run by the LPG gas. Okay, after 5 hours distillation, the oil was extracted from the leaves of mint. Okay, so but for the last experiment which we perform on 1000 square meters area, the biomass very much huge. Then we harvested the whole biomass from the field of 1000 square area, and then we carried out this biomass to the mint factories, okay, menthol factories. The whole biomass was dropped in a very huge container. Okay, these huge containers are filled with this biomass, and then the lid was covered, water was filled, and run for 5 hours. The oil was extracted by this technique. And you will be surprised to know, in case of control plant, we obtained only 4.73 kg per 1000 square meter area of the land, but in case of treated plant, we find 7.56 kg in 1000 square meter area. Okay, so in this way, about 60% increase was obtained. I represented by report at BRC, and all the scientists were very much happy, and Dr. Vashni is also working that how to get it patented, this technique. Okay, so not only the oil content but also menthol content and essential content was also increased by this treatment. So after carricane and after RPPs, we also work on tricontrol. This is a long chain alcohol. This is also growth promoter available in the market. Okay, this has also a miraculous effect, and we did a lot of plant. And then the fourth plant growth is new one. This is known as MDM, methyl alpha D manoperoxide. So this company is in USA. I wrote a letter to them that I am working on medicinal plant. None of you did we applied this chemical on the medicinal plant, so we want to check it, that whether this chemical can also work on medicinal plant. So they made a meeting and they granted 1 gallon of this MDM and sent to us. They say that by the application of MDM, the cherry yield can be increased by 10%. Okay, this is the company name, branded Hammer. And in case of watermelon, 26% increase was done. In case of tomato, 21% increase was increased. Okay. In case of cantaloupe, खरबूजा से कहते हैं, 12% increase was done. In case of lettuce, salad, 24% increase was done. In case of pepper, Shimla mirch, 11% increase was yield. So this was the report by the branded Hammer. So then I wrote to them that if we apply this chemical on bristle plant, can we increase the growth and yield and quality of plant? So they were happy and they granted a gallon of this chemical. Blueberry, ये बहुत full of antioxidant, बहुत ज़्यादा इसमें antioxidant होते हैं, and very expensive also, 26% increase. In case of cabbage, 7% increase was done. So then they gave this MDM to us, and the mechanism of action of this is that when the Calvin cycle is over, millions of glucose molecules are synthesized, as you know very well, right? So many of the glucose molecules are trapped by plant proteins known as lectins. Okay, lectins trap thousands of glucose molecules, and when those glucose molecules are trapped, they are useless for plant. They can't help plant in their growth and yield. So what do they say? That their chemical MDM goes to the lectin where the glucose molecules are bound, and they the MDM says that get out from here. So the MDM is fixed over here, which is a non-toxic compound, and the glucose molecules are released. When these glucose molecules are released in the plant, they are utilized in different metabolic phenomena like respiration and this and that. Okay, so this is the concept, their concept, that MDM releases the glucose molecule which are trapped by plant lectins, plant proteins, and when the glucose molecules are released in cytoplasm, this is utilized in many constructional work in the plant cell, right? So we did this for on curcuma longa. हमने हल्दी पे काम किया इस पे यही डिपार्टमेंट में, and we found out that fresh weight, dry weight, chlorophyll A, chlorophyll B, carotenoid, electron transport rate, phenol content, flavonoid content, and leaf thickness, stem diameter, curcumin content was also increased by the application of this new chemical, new PGR, known as MDM methyl monoside. So they said that this chemical is harmless, and this releases the glucose molecule which are unfortunately trapped by the plant proteins lectins, and when these glucose molecules are released from the plant proteins, okay, they are utilized in different metabolic phenomena in the plant, and in this way this can increase the plant productivity. So in this way we did a number of work on different plant. So this is the paper which published on Carikin. I have also brought some copies of this paper, and you can take one copy for each of you. आप लेक्चर के बाद यहाँ रखे हुए हैं, एक कॉपी ले लीजिएगा, ठीक है? Okay. Okay. Now this is over. If you want to ask anything, I will try my best to reply, whatever I know. If you want to give any suggestion, any question, you are welcome to do so. ठीक है? You can ask any time. ठीक है? And also I will try to show you this smoke generating apparatus, and maybe tomorrow or day after the organizers give me a slot of about half an hour. I will show you that how it works, right? We are working on this, and if the time permits, I will show you the whole apparatus, how does it work, right? ओके, ठीक है साहब, आइए।
Delivering an very elaborate talk on such pertinent topic of this course. So hope you might have enjoyed this novel findings which Professor Masroor has shared with you, and you just try to incorporate in your future programs by just listening his lecture. So thank you Professor Masroor Sahab for giving time and delivering lecture. Thank you very much. So, welcome again to all of you. We are assembled here to just go into details of very interesting lecture that is likely to be delivered by Andrzej on the topic challenges and practical problems of plant hormones in biotechnology. So, Andrzej, you are again welcome to deliver your new lecture. Thank you.
Thank you very much. I hope that lecture will be interested. Yep. But I hope that after lunch you have more power to sit and hear my lecture. The subject, of course, will be challenges and practical problems of plant hormone in, maybe hormones in biotechnology. So I will welcome again. Let's start, of course, as biotechnology advances, we have gained incredible insight into phytohormones applications. Of course, from tissue culture and genetics engineering to crop improvement and post-harvest technology. However, the journey is not without its challenges. The practical application of plant hormones in biotechnology face numerous challenges as difficulties in large-scale production, instability under environmental conditions, and of regulatory concern. Additionally, hormonal crosstalk, the complex of interactions between different hormones makes precise control difficult. These challenges demand innovative solutions that integrate biotechnology, nanotechnology, and advances analytical techniques. In this lecture, I would like to present some other waste contaminants dangerous for us, for our planet. That's mean nanoparticles, micro and nanoplastic. Of course, with correlation to plant hormones. Let's begin this fascinating journey in the world of plant hormones again, and now biotechnology. We know that phytohormones are chemical messengers of plants orchestrating growth, differentiation, and response of environmental stimuli. In biotechnology, they significant cannot overstand it. They serve as key drivers in several crucial fields. That mean tissue culture and micropropagation, where phytohormones are essential in callus induction, organogenesis, somatic embryogenesis, the core techniques of plant tissues culture. By carefully manipulating hormones like auxin and cytokinins, we can regenerate entire plants from a single cell, enabling large-scale propagation of available plant species. The next, of course, key is genetics engineering. It means that modern biotechnology allows us to alter plant hormone biosynthetic pathways, leading to crop with enhanced growth, improved stress tolerance, and high yields. By targeting genes responsible for hormone production, we can design plants that are more resilient to environmental stresses. And third, crop improvement. Through hormone regulation, we can increase productivity and stress resistant in crop. For example, gibberellins can boost plant hay and seed germination, while abscisic acid helps plants survive extreme drought conditions. The next secondary metabolite production were many medical for industrial plants rely on phytohormones to synthesize bioactive compounds. By fine-tune hormone levels, we can enhance the production of available secondary metabolites used in pharmaceuticals, cosmetics, and food industries. So, if post-harvest technology, even after harvesting, phytohormones continue to play a role. They regulate ripening, senescence, storage, quality, allows us extend shelf life, reduce food waste, and improve market value. This application illustrates that how hormones bridge the gap between fundamental plant physiology, plant biology, plant biochemistry, and applied biotechnology. However, while the potential is enormous, the practical application is met with significant challenges, from production to stability issues to regulatory concern and unintended physiological effects. The next slide is, I suggest, I would like, maybe not suggest, I would like to omit, because sometimes I talk and show these challenges, more knowledge about phytohormones, which I present in the previous some lectures. I would like to show that the modern environmental threats include nanoparticles, microplastics, and nanoplastics which persist in ecosystem, disturb soil, water quality, and pose risk to plant, animal, and human health. These contaminants can alter plant growth, interfere with nutrients uptake, and affect hormonal signaling pathways. Phytohormones offer a potential solution by enhancing resilience against these pollutants. They regulate stress response similar like other contaminants, improve nutrients absorption, mitigate the toxic effects of these very dangerous and newest contaminants. Additionally, research onto phytohormone-based strategies such as hormone-assisted phytoremediation aim to enhance plant detoxification mechanism, providing a sustainable approach to tackling pollution. Our understanding that hormone nanoparticle or micro-nano-plastic interactions grow, phytohormones may become a key important tool in reducing environmental damage and promoting sustainable agricultural practice in the world increasingly affected by these pollutants. We know that nanoparticles are materials with at least one dimension between 1 and 100 nanometers. But why are nanoparticles relevant to plant biology and phytohormones? Their small size allows them to interact with plant cells at the molecular levels, influencing growth, stress response, and hormone regulation. Some nanoparticles can mimic phytohormones, while others can enhance hormone delivery, stability, and efficiency in plants. We must know that, for example, ions, metal, can convert into nanoparticles. For example, silver ions can convert into silver nanoparticles in chlorella vulgaris, which was a part of my team study. However, despite their potential, not all nanoparticles interacts are beneficial. Some nanoparticles accumulate in plant tissues, potentially leading to the toxicity and give oxidative stress and hormonal imbalance. Nanoparticles can be categorized on the composition and origin. Each type will exhibit distinct properties that influence their application in biotechnology, medicine, and agriculture. For example, there are metallic nanoparticles, metal oxide nanoparticles, carbon-based nanoparticles, and polymeric nanoparticles. Use, for example, are biodegradable nanocarriers made from natural or synthetic polymers. They usually used for controlled release of nutrients, pesticides, and phytohormones, ensuring long-term efficiency in minimal environmental impact. We know that nanoparticles can origin from both natural and anthropogenic sources, with natural source often resulting from natural processes, anthropogenic sources stemming from human activities. For example, natural sources. Nanoparticles can be formed from volcanic eruption, forest fires, and soil erosion. Anthropogenic sources are following: from factories, vehicles, and house, and agricultural application are significant contributors to these nanoparticles. Agricultural practice, such as use of nano-fertilizers and nano-pesticides, contribute to increase presence of nanoparticles in soil and crops and water. Plastic products, as they break down, release microplastic, nanoplastic. It's dangerous, though. This sources highlight that nanoparticles are not just a byproduct of industry's process, but also an inevitable presence in nature. Nanoparticles have shown significant promise in several key areas of agriculture, with application ranging from enhanced nutrient delivery to enhanced pest control and even genetics formation. For example, nano-fertilizers for enhanced plant growth. As example, zinc oxide, iron oxide, or titanium dioxide serve as carriers for micronutrients, enhancing the viability and reducing nutrients leaching. The advantage of using nanoparticles lies in the slow release properties, which prevent nutrients loss and enhance uptake efficiency over time. For example, zinc oxide nanoparticles can improve zinc uptake, which is essential for immune responses. How they contribute to agricultural biology as nanoparticles and nano-herbicide. They can also use to control release of pesticide and herbicide, reducing toxicity and minimizing environmental conditions. For example, silver nanoparticles exhibit antimicrobial properties, preventing fungal and bacterial infection in crops. We know that silver nanoparticles may exist in deodorant, in perfume. Stop the odor. Chitosan-based nanoherbicides selectively target weed growth, offering a more precise and environmental friendly approach compared to the traditional herbicide. This example illustrates that potential of nanoparticles to improve both crop productivity and environmental sustainability. However, as with any innovation, there are risks and challenges. For example, long-term accumulation of nanoparticles in the soil, their interaction with plants, and their potential toxicity to beneficial soil microbes remain concerned that need careful evaluation. How nanoparticles interact with soil, water, and air. Understanding this interaction is important for assessing the long-term sustainability of using nanoparticles in agricultural biotechnology. Nanoparticles can have a significant influence on soil structure and nutrient cycling. For example, they can improve water retention, which is particularly important in drought-prone areas. On the other hand, they can also alter microbial communities in the soil, potentially affecting plant growth and soil fertility. In some cases, nanoparticles may accumulate in the soil, leading to toxicity for soil-dwelling organisms. This disruption can have cascading effects on nutrient cycling, which in turn may affect plant health and productivity. In water, nanoparticles, especially those derived from nanopesticides and fertilizers, can leach into water system, potentially contaminating rivers, lakes, and groundwater. Why some nanoparticles, such as iron oxide nanoparticles, have been explored for their ability to remove contaminants from waters, and others may have harmful effects on aquatic organisms. For example, silver nanoparticles can exhibit toxicity on aquatic organisms. For example, Chlorella vulgaris algae, which I studied. Furthermore, nanoparticles in water system may bioaccumulate in this organism, in aquatic organism, plant, of course, in animals, eventually entering the food chain, which can lead to human health conditions. What about in air? Nanoparticles are also present in atmospheric aerosols, contributing air pollutions. They can travel long distance through the air and settle on soil, plants, and water bodies of many organisms, where they can have various effects on plant growth and, of course, ecosystem balance. Some studies suggest that the airborne nanoparticles can interfere with plant photosynthesis and growth, especially if they accumulate on plant surface, blocking the stomatal pores and preventing gas exchange. While nanoparticles offer tremendous potential in agriculture, their presence in environmental raises critical concern. How nanoparticles interact with plants? The most common pathways for nanoparticles to enter is through root or absorption. Often absorbed nanoparticles travel via the xylem to the shoots and the leaves. The nanoparticles are delivering essential nutrients such as zinc or iron, or if they're enhancing plant resilience to stress. Example, iron oxide nanoparticles can improve iron uptake, which is crucial for photosynthesis and overall plant health. Nanoparticles can also be absorbed through the foliage, through the stomata and cuticular layers. This route of uptake is particularly used for the delivery of phytohormones or other bioactive molecules directly to the plant tissues. Titanium dioxide nanoparticles have been studies to the ability to enhance photosynthetic efficiency when applied to leaf surface. They can be beneficial under high light stress, where they help optimize light absorption and reduce photooxidative damage. Some nanoparticles are known to modulate plant hormone signaling pathways, particularly those related to abiotic stress tolerance. For example, silicon on iron oxide nanoparticles have been shown to prove plant growth tolerance to drought and salinity by enhancing abscisic acid signaling and reactive oxygen species scavenging. Nanoparticles have been a significant impact, such as positive and negative on various physiological processes. The first, of course, the positive effects on plant physiology. Nanoparticles, especially zinc oxide or iron oxide, serve as carriers for essential nutrients, improving their ability and absorbing by plant roots. Nanoparticles like silicon nanoparticles and iron oxide may enhance plant resilience against abiotic stresses. They help by manipulating plant hormones, for example, abscisic acid, which play, of course, a crucial role with drought tolerance and stomatal regulation, on which you better know. Some nanoparticles, like gold nanoparticles, can promote cell division and root elongation by interacting with cytokinin pathways. They make useful in application like tissue culture or plant regeneration. The negative effects on plant physiology. The high concentration of nanoparticles can generate reactive oxygen species, leading to oxidative stress, on which I talk about this. The stress damages cell membranes, protein, and DNA. Some nanoparticles can disturb normal plant hormonal regulation. For example, silver nanoparticles have been found to interfere with oxygen synthesis, leading to reduced root elongation. It similarly act zinc oxide with gibberellin biosynthesis, resulting to stagnant growth. High concentration of nanoparticles, especially those that accumulate on a leaf surface, can block stomatal pores, interfering with gas exchange and photosynthesis. As you can see, nanoparticles have a potential to both enhance and impair plant physiology, depending on factors such as concentration, type of nanoparticles, and the plant species being studied. This underscores the importance of carefully managing nanoparticles application to avoid potential toxicity and environmental harm. How nanoparticles affects on plant tissue and plant tissue cultures and genetic engineering. One of the one of the major challenges in genetic engineering. Of course, nanoparticles such as gold nanoparticles or carbon nanotubes have been used as a carrier for DNA and RNA molecules. And these nanoparticles can penetrate plant cell wall more easily and facilitate the uptake of genetic materials into plant cells, enhancing transformation efficiency. The use of nanoparticles allows non-vital gene delivery, which is often safer and more efficient than traditional methods such as agrobacterium-mediated transformation on gene guns. This is especially important when working with special species that are difficult to transform, or even more precise control methods of genetics modification is still required. The next polygylation, pegylation, is a process of both covalent and non-covalent attachment or amalgamation of polyethylene glycol, and silicon nanoparticles are another example. This nanoparticle has been shown to improve the efficiency of CRISPR-Cas9 gene editing system, enabling more precise and targeting modification in plants. On the other hand, magnetic nanoparticles are also gaining attention for their ability to assist in vitro DNA uptake during plant tissue engineering. The use of magnetic fields can help guide nanoparticles directly into plant cells, making gene editing faster and more precise. The interaction of nanoparticles and phytohormones can either enhance or inhibit the plant's natural responses. For example, silver nanoparticles can enhance salicylic acid-mediated defense mechanism, improving the plant's ability to resist fungal and bacterial infections. This is particularly useful in combining plant disease, as salicylic acid plays a critical role in systematic acquired resistant SIR. Titanium dioxide nanoparticles can increase photosynthetic efficiency by enhancing cytokinin activity, leading to improved cell division and shoot regeneration, and this results in increased plant growth under certain conditions. Silicon nanoparticles have been shown to modulate abscisic acid signaling, improving plant ability to tolerance salinity and drought stress. Crosstalk between nanoparticles and phytohormones highlights the delicate balance a plant must maintain to regulate their growth and response to environmental stresses, while nanoparticles can boost stress resistance and enhance growth. They can also disturb hormonal pathways, leading to unexpected physiological effects. Is of course dangerous. It's equally important to consider the impact on human and animal health, especially when nanoparticles are used in agricultural practices and can enter the food chain. For example, inhaled nanoparticles may cause pulmonary inflammation, leading to lung disease, such as asthma. Some nanoparticles, due to their small size, have the potential to cross the blood-brain barrier, possibly affecting neural function and leading to cognitive impairments. Nanoparticles such as zero-valent iron nanoparticles are heavily effective at removing heavy metals like arsenic, lead, cadmium from contaminated soil and groundwater. These nanoparticles are the source pollutants and facilitate chemical reactions, then neutralize them. Graphene oxide nanoparticles have been shown to remove organic pollutants such as pesticides or pharmaceuticals from wastewater by acting as adsorbent, binding to harmful substances and reducing their bioavailability. Nanoparticles play essential role in improving water purification process and application of nanoparticles for pollution cleanup presents on opportunity to restore ecosystem affected by industrial pollution, agricultural runoff, and mining activities. However, it's important to remember that while nanoparticles can enhance remediation efforts, the long-term environmental impact must be studied. Now, I would like to shift our focus on very promising development in the field of nanotechnology, the use of biodegradable nanoparticles, which I would like to underline, in eco-friendly application. As we continue to face growing concern about the environmental impact of traditional nanoparticles, there is increasing interest in using biodegradable alternatives that breaks down naturally, reducing the long-term presence in our ecosystem. These nanoparticles have made from materials like chitosan, alginate, and cellulose, represent a major advancement in sustainability. Those nanoparticles are naturally degradable, meaning they break down into harmless byproducts over time, ensuring that they do not accumulate in the environment, what is once again, very important. For example, chitosan nanoparticles, which are derived from crustacean shells, have gained attention for their biodegradable nature and antibacterial, antimicrobial properties, making them very useful in agricultural application as biopesticides. The next herbivores, such as insects, can ingest nanoparticles when they mosquitoes. I forget spray repellent. Okay, the herbivores can ingest nanoparticles when they feed on contaminants plants. This can affect their growth, metabolism, and immune system. And nanoparticle can also bioaccumulate in higher trophic levels, meaning that predators who consume contaminated herbivores may experience higher concentration of nanoparticles in their bodies as a phenomenon known as magnification, biomagnification, of course. This process can disturb ecosystem stability and lead to potential health problem for animals and for us. At the top, the food change, and the top change, food change. The long-term persistence of nanoparticles in the environmental raises questions about their potential impact on the ecological balance. Over time, accumulation in soil, water, and plants can disturb an entire ecosystem, affecting microbial population, plant diversity, and animal health. In particular, nanoparticles may have unknown ecological consequences, as the long-term effects are still not fully understood. This calls for careful monitoring and regulation of nanoparticles use in agricultural and environmental application. As I remember, I previously presented information concerning the interactions of phytohormones with nanoparticles. So I will skip some slides, but I would like to summarize. One of the most promising benefits, of course, skip a few slides according to your printer version of my presentation. You have my printed version, I hope. No? But I present the same or similar information about interaction of phytohormones. It will be borrowed for you and not me, but for you. Okay, one of the most promising benefits is a promotion of plant growth through the interactions between nanoparticles and phytohormones. For example, nanoparticles can influence hormonal pathways that can regulate auxin, cytokinin, gibberellin levels. Example, silicon nanoparticles can enhance root elongation via auxin up regulation, and gold nanoparticle can stimulate root regeneration by enhancing cytokinin activity. This interaction helps accelerate plant development, making nanoparticles an ideal tool for improving crop production. Another important benefit is the ability of nanoparticle to enhance plant resilience under stress conditions. We know that nanoparticle can modulate the signaling of abscisic acid, salicylic acid, which are involved in stress responses. Of course, that while a nanoparticle can enhance plant growth, excessive exposure can lead to hormonal imbalances. High concentration of nanoparticles can disturb the synthesis and transport of phytohormones, leading to abnormal growth patterns or toxicity. Yes, there's. Oh. The interaction between presents both exciting opportunities and significant challenges. By carefully controlling the dose, the type of nanoparticles and specific phytohormones involved, we can harness the positive effects on plant growth. So, as we can see in table, nanoparticle can enhance the effects of phytohormones, and nanoparticles improve the plant response to abiotic stressors. Nanoparticles have been found to enhance plant immunity, particularly through the mediation of salicylic acid and osmonic acid, offering improved resistance against pathogens. However, they use the high concentration of metal-based nanoparticles may result in oxidative stress and cell toxicity, which could counteract these beneficial effects. Smart nano fertilizers hold the potential for reducing agrochemical waste, and the target delivery of nanoparticle can improve crop yield with further inputs, benefiting food production efficiency. As I conclude my presentation on the interactions of nanoparticles and phytohormones in biotechnology, yep, many is
Reprint? Mosquito reprint you need?
no. Reprint?
You need mosquito reprint?
No, reprint, no, I can't. No, I have. I bought very strong repellent against mosquitoes, especially tropical mosquitoes. And I can use this repellent, this muga, for domestic mosquitoes. I killed. They are dangerous for me because I'm afraid Plasmodium vivax. So I must every day take special tabs against this pathogen. Yeah. Okay. What about long-term studies, which are essential for understanding the bioaccumulation of nanoparticles in plant tissues and their potential, of course, effects? Some challenges, of course, I would like to present. Researchers must focus on how nanoparticles move through the food chain and the long-term impact they could have on the soil health, water system, and organisms that really on plant for food. The studies will help us identify safe concentration and application methods to minimize risk to human health and the environment. In conclusion, while nanoparticles offer immense potential to improve agricultural productivity and plant resilience, it is crucial to carefully manage their application to avoid any unintended consequences. By pursuing biodegradable options of nanoparticles, optimizing nanoparticles, nanoformulations conducted in deep studies and complementing strict regulatory frameworks, we can maximize the benefits of nanoparticles while minimizing their risk. The next challenge which we can find in environment are nano or micro or nano plastics. Much like nanoparticles, have raised growing concerns due to their widespread presence in the environment and then potential to affect plant growth, development and stress responses. Again, the same. Microplastics, typically defined as plastics particle, ranging from 1 micrometer to 5 millimeters in size, and nanoplastic, which are even smaller and range from 1 nanometer to 100 nanometers, have become pervasive in our ecosystem. The sources of these particles can be divided into main categories of primary sources and secondary sources. The primary sources refer to plastic that are intentionally produced at smaller and the smallest scales. For instance, we often encounter micro beads used in personal care products such as toothpaste and facial scrubs. These tiny particles, through effects in their intended use, contribute to the growing plastic pollution problem when wasted down drains. Secondary sources of microplastics and nanoplastics are even more concerning, as they originate from the breakdown the larger plastics item. Over time, every day, plastic products, whether from plastic bags, bottles, and packaging, degrade due to factors like ultraviolet radiation, mechanical abrasion, and microbial activity. These larger plastic materials break into progressively smaller pieces until they became micro and nanoplastics, dispersing widely throughout the environment. One of the most significant concerns is contamination of soil. As microplastic and nanoplastic enter the soil, they affect soil structure and water retentions. They can disturb essentially soil microbial communities, which play a crucial role in the nutrients cycling, soil fertility, and plant growth. Microplastic and nanoplastic also pose a significant threat to water quality. When plastic particles enter rivers, lakes, and oceans, they can be ingested by aquatic organisms, including fish, marine invertebrates, plants, and others, causing physical harm or by accumulation of toxins. This plastic can also disturb marine ecosystem by clogging the respiratory system of aquatic organisms, reducing their ability to absorb oxygen. One of the most critical impact of microplastic is on microbial communities and soil fauna, both which are vital for the health ecosystem and sustainability of agricultural systems. We know that microplastic enter the food chain through several pathways. So I would like to go. Microplastic or microplastic and nanoplastic interfere can alter, of course, hormonal pathways in plants. They interfere with the transport of auxin and cytokinin, which stop. Additionally, for example, polystyrene nanoplastics disturb abscisic acid regulation, and plastics have ability to absorb phytohormones, which reduce their viability in the soils. A case in this point is abscisic acid, which binds to microplastic particles, those becoming less available for plants to respond to drought stress. The interaction complicates the way plants manage environmental challenges, emphasizing the need to understand this mechanism better for efficient agricultural practice. Microplastic and nanoplastic are not just passive pollutants. They act as a carrier to various chemical substances, including metal nanoparticles and agrochemicals. For instance, microplastic can absorb pesticide, which can interfere with biosynthesis of gibberellins. When plants are exposed to this in the signaling pathways, their growth and development may be compromised, affecting crop yield and health plants. This is some challenge. The first is the accumulation in agricultural zone. The second is the second risk concern the potential contamination of crops. Studies shown that microplastic and nanoplastic can be found in crops, including common staples like wheat, rice, lettuce, and carrots. If these particles enter the food chain, they can have long-term environmental and health impacts, so particularly through the consumption contaminated produce. Finally, of course, we face regulatory challenges and the lack of awareness. Currently, there are not global standards for monitoring microplastic pollution. It's a pity, of course, in agricultural soil, and many farmers and consumers remain unaware of the potential risk posed by these particles. This gap in knowledge highlights the need for more resistance and clearer guidelines to address the growing issue of microplastic contamination in agriculture. In the slide I summarize, I revisit the key point regarding the benefits and risk associated with microplastic and nanoplastic in agricultural system. The risks are very significant, particularly concerning the accumulation in the environment. Microplastic and nanoplastic do not degrade easily, leading to persistent environmental pollution. And what else? This particle can also enter the food chain, and it's of course, therefore, is crucial between balance and this benefits and risk. Effective solution should be developed to minimize the negative impact, which is shown on the right column, negative impact of microplastic and nanoplastic, while capitalizing on the potential advantages. Of course, the future research and regulation will play a vital role in finding these balances. As we look towards the future, there are several important solutions that can help address the challenges posed by microplastic and nanoplastic and agricultural. First, there is a need to development biodegradable plastic to replace traditional, for example, petroleum-based plastic. The second is improving filtration and removal technology is crucial, especially in wastewater and agricultural runoff. Another critical step is to regulate plastic use in agricultural, particularly reducing reliance on plastic-based mulches and fertilizers. And lastly, it is essential to promote public awareness and conduct more research on the long-term effects on nanoplastic, microplastic, nanoplastic pollution. Understanding, of course, this impact is a key to developing informed policies and solutions that protect ecosystem, human health, and agricultural system. Of course, another summary of challenges. One of the primary challenges is difficult in a large-scale production and extraction, for example, of phytohormones. Extraction is very easy in the large-scale production sometimes. These challenges are compounded by the instability and degradation of phytohormone, as they are highly sensitive to environmental factors such as light, temperature, and pH. Their rapid metabolism and breakdown in plant tissues complicate their use in biotechnology applications. Another challenge is the crosstalk between different phytohormones, which can result in unintended physiological effects. Meaning this interaction is key for precise hormone control in plant system. Analytical challenges also persist, so current detection methods require very high sensitivity and advances technical expertise. We know about LCMS, MS techniques. Maybe another go. Oh, in short, thank you very much. This is the last. Not the last. No, sorry, not the last. My last, the last. Yes. Yes. I must thank you.
and today we will explore the topic that is regulation of nitric oxide, that is role in agriculture. So, how many of you know about the nitric oxide? Nitric oxide is a fascinating molecule that has gained significant attention in recent years due to its diverse role in both animals and plant physiology. It is not only important for the plant, but it is also equally important for the animal physiology also. And in this presentation, we will explore the biological functions of nitric oxide, its synthesis, regulation in plants and its potential applications in agriculture. So, first of all, moving forward to the discussing the all details, we have to know what is the nitric oxide. So, nitric oxide is a small, highly reactive and diffusible molecule that act as a signaling agent in biological system. That is why it is very important for the both animals and plants. It was initially considered toxic due to its presence in industrial waste and exhaust fume and associated with the acid rain and air pollution. But however, in the earlier 19th century, researchers discovered its critical role in regulating the human cardiovascular system and leading to the paradigm shift in our understanding of nitric oxide. And for this work in 1992, NO was named as the molecule of the year, and in 1998, 3 American scientists, F. Murad, Fugard and Ignaro were awarded with the Nobel Prize in the work of NO. So, if we see what are the properties of nitric oxide that making it the molecule of the year. So, the unique properties of NO that make it versatile molecule in biological system, that is its lipophilic, meaning it can easily cross the cell membrane, and it has an unpaired electron that make it highly reactive. As you can see in the structure that NO has a one unpaired electron that making it highly reactive in nature, and this reactivity allows NO to interact with various molecular targets including metals and thiol containing proteins. So, additionally NO react with reactive species and forming the reactive nitrogen species. So, all of you know the ROS and RNS both are very important and used as signaling molecules at very low level. But when their level is exceed, they are working as they create the aging and many processes that call the stress responses. So, at the cellular level, when their condition is at the optimum level, it is very crucial and it work as a signaling molecules. So, NO have emerged as a signaling molecule due to their prominent existence and diverse role in cellular metabolism, signaling pathways and induction of oxidative stress, regulation of stomatal guard cell opening and modulation of plasma membrane fluidity, and contribution in programmed cell death at low concentration. So, it is very important to know at all these processes involved in low concentration. However, if the amount of this NO surpasses a certain threshold level, they have the possibility to trigger the damage to the cell and that causes the cytotoxicity. So, that is why the topic of my talk is the regulation of nitric oxide, because if it exceed is called reduce the plant cytotoxicity. At lower level it enhances the many biological functions. So, our focus in this talk is on the regulation and beneficial aspect of nitric oxide, not the harmful aspects. That is why I am not going to discuss about the RNS, that is the reactive species. I will discuss only the beneficial aspect of nitric oxide. So, if we see the regulation of nitric oxide, there are 3 main pathways that impart the regulation. The first one is the cellular homeostasis that is achieved by the synthesis and scavenging, and there are some key regulatory mechanisms include the interaction with reactive species and processes like S-nitrosylation and denitrosylation, and further NO is degraded by some enzymes like NO deoxygenase that binds with and it also binds with the antioxidant like glutathione. So, that is why antioxidant is also very important for the plant as well as for the human system as well as for the animals, because it binds with the excess oxidants जो भी generate होंगे हमारे body में, that binds with these oxidants. So, if we see the NO homeostasis, it is essential for signaling functions in plants, and the equilibrium between this NO synthesis and scavenging determines the endogenous levels of nitric oxide. which must be carefully balanced to respond to various physiological and environmental stress conditions. And this balance ensures that NO can effectively regulate plant growth, development and stress responses. And for this, uh, first of all, we have to discuss what are the sources and synthesis of nitric oxide in plants, because NO homeostasis depend upon the two things, its synthesis and its scavenging. So, what are the sources of NO in plants? So, in plants, the primary source of nitric oxide is through the reduction of nitrite, that primarily occurring via the enzyme nitrate reductase that located in the cytosol and plasma membrane. With other potential sources include mitochondrial electron transport, xanthine dehydrogenase and the NOS pathway, which was first discovered in animals, but now it is also reported in plant system. And the other pathways of nitrosynthesis, NO synthesis is the non-enzymatic pathway that under the acidic conditions, however the exact mechanism of NO production in plants are still being researched, it says that the under acidic condition NO is simultaneously produced from the nitrate. So, there are the multiple pathways of NO which we are going to discuss one by one. So, this biosynthesis include mainly two pathways. First one, that is the enzymatic, and the second one is the non-enzymatic. There are several enzymes which involved in the synthesis of nitric oxides. So, first of all, what are the biosynthetic pathway that due to the enzymatic activities? So, the key enzyme pathways include the NR pathway, NOS pathway, xanthine oxidase pathway and mitochondrial electron transport chain, and the first one in this the nitrate reductase pathway. This NR reduces the nitrate to nitrite, which can further convert it to NO, and this pathway is crucial at under low oxygen condition. And the second one is the nitric oxide synthase like activity. This enzyme, although a homolog to animal NOS and has not been identified in plants, but its like activity has been reported and utilizes the arginine to produce the nitric oxide in plant system. In similar way, the next enzyme, that is the xanthine oxidase, that generate the nitric oxide through the reduced nitrite to NO under the hypoxic conditions. And the last one in the enzyme activity is the polyamine oxidase pathway. In this, oxidation of polyamines such as spermine and spermidine can lead to the NO production through the reduction pathway. So, if we see, these are the enzymatic pathway, and the second one is the synthesized by the non-enzymatic sources, and these are the basically reduction of nitrite to NO can occur in the presence of reducing agents like ascorbates and phenolics. So, these are the sources of non-enzymatic, and it is also generated at low pH. At acidic pH, nitrite can simultaneously convert it to NO. So, enzymatic pathways involve the enzymes and NO simultaneously generated at acidic condition, where it can also be reduced through the ascorbate or phenolic through the in the non-enzymatic sources. So, if you see the overview of the nitric oxide biosynthesis, this is the production of NO from different enzymatic and non-enzymatic pathways that includes the NOS, that is nitric oxide synthetase like activity, NR, that is the nitrate reductase activity, and NIR. In this pathway, you can see NO is synthesized from the different pathways, and the main in this the NR pathway. That is, here you can see nitrate, conversion of the nitrate, then this NO, nitrate from this converted into the NO from the here. So, the production of NO from different enzymatic, all this represented here. Beside this is the xanthine oxidase production pathway, and in the mitochondrial electron transport chain NO is also produced. So, overall it is produced by mainly the enzymatic pathways, and there are some sources of non-enzymatic production of nitric oxides. So, these are the mainly biosynthesis pathways of nitric oxide, and here these pathways are further categorized into two categories depending upon the which type of reaction is going on, and these are the either oxidative pathway or reductive pathway. Most of the enzymatic reaction are categorized under the reductive pathways, and all the different as shown in this one, these are the oxidative pathway, that is NO generated from the either arginine or polyamines or hydrogen. These are the oxidative pathways of NO generation. So, the reductive pathway, this pathway involves the reduction of nitrogen containing compounds to produce NO, and the key enzyme as we have discussed earlier is the nitrate reductase, xanthine oxidase, and It also includes in the reductive pathway, that is the non-enzymatic reduction, that is happen simultaneously through the at the acidic condition and through the ascorbate or phenolics. While in the oxidative mechanism, this pathway involves the oxidation of nitrogen containing compounds to generate NO, and the enzyme in this include the NOS pathway as we have discussed earlier, and the polyamine oxidase pathway. So these are the two main mechanism, enzymatic and non-enzymatic, and these are the two pathways from the oxidative and reductive depending upon the reaction, which type of reaction is taking place to generate the NO. So these are the biosynthesis of NO taking place. Then the next one, this is the biosynthesis, and the next for the homeostasis, maintaining the balance of the NO, that is the scavenging is also very important. As I have told you earlier, the balance of NO is very important because at low concentration it incorporates to the cellular signaling, but at higher concentration it can cause the cytotoxicity. So homeostasis is very important, and homeostasis is maintained by the scavenging of different pathways, and in scavenging of the plant is mediated by the several pathways including the phytoglobins, S-nitroglutathione reductase activities, and thyroidexin. These are the three main pathways of scavenging of nitric oxide, and in plants this nitric oxide is scavenged by the also reactive oxygen species and by hemoglobins. So these are the some pathways of nitric oxide scavenging, in which one of the main important is that's a phytoglobin. Phytoglobins are plant hemoglobin that scavenge the nitric oxide. Hemoglobin oxidizes the nitric oxide to nitrate, and the non-chemotic hemoglobin is involved in the oxidative degradation of NO to produce the nitrate in the first case, and in the second one, by the second, that is the S-nitroglutathione reductase enzyme, which regulate the S-nitrosylation, that is the post-translation modification mediated by NO. So this thiol binds to NO and transport it to the site of action, and where the nitrothiols release NO after the intercellular diffusion and uptake into the target cells. So this N-nitrothiol glutathione is an NO reservoir and a long distance signaling molecule for the NO homeostasis. So this GS-NO, it also is scavenged as it, and also it is storing it when it requires, it process to the different locations of the cell. And the third mechanism which we have discussed earlier, that is the thyrodoxine H5, which act as a direct denitrosylase and helping to maintain the NO homeostasis. And this equilibrium between the NO synthesis and scavenging mechanism determines the plant endogenous levels of NO, and this homeostasis is necessary for the NO to exhibit its signaling functions. So all the functions of NO depending upon this levels of NO, and it plays a crucial role in plant responses to drought, salinity, and heavy metal stress. And this is the outline which we have studied in the biosynthesis and scavenging, that is the total metabolism of nitric oxide in plant cells. Nitric oxide can be generated among others by either nitrate reductase or by L-arginine dependent nitric oxide synthetase. Then NO can react with the reduced glutathione to form N-nitrosoglutathione through a process of S-nitrosylation, and the metabolite can be converted by the enzyme S-glutathione and reductase into oxidized glutathione and ammonia. Then is further this glutathione which make mediated signaling processes, and ultimately the nitric oxide also interact rapidly with the superoxide radical to generate peroxynitriles and a powerful oxygen molecule that can mediate the tyrosine nitration for the proteins. So in this process you can see this is all our metabolism of nitric oxide, how it is synthesized and how it is mediated signaling responses of the plant, as you can see in this particular figure. So synthesis takes place, then its post-translation modification, and then it causes the cell signaling, which is important for the different biological functions of the plants. And it regulates many growth and development processes from growth seed germination to the senescence. So it impart many growth regulation processes. So how this signaling is taking place? So NO, as I have discussed earlier, NO is a very diffusible molecule and reactive. So it can diffuse freely across the cell membrane and interact with various cellular targets including proteins, causing post-translation modifications like S-nitrosylation, and which alters the protein functions and triggers the downstream signaling pathways. So this one is the reaction, that is the S-nitrosylation is very important, that triggers most of the signaling process by the NO. Besides this, NO often interacts with other signaling molecules like reactive oxygen species, modulation of calcium signals, protein modifications, and phytohormones, hydrogen sulfide, and creating a complex signaling network that regulate most of the developing process of the plants. So it's not only act as a signaling molecule, but it also crosstalk with other signaling molecules to regulate the different functions of plants from germination to senescence. So NO targets various molecules including enzymes, transcription factors, and DNA that leading to the post-translation modifications that regulate the plant growth, development, and stress responses. So here in this figure, a schematic representation of nitric oxide signaling in plant cells is presented, and that interplay between the nitric oxide, calcium, and protein kinases in plant cells. So the direct NO signaling mechanism, there is a presence of S-nitrosylation, which we have discussed earlier. Beside this, it is also binds with the other metal NO interactions, like NO directly bind to the metal centers in heme containing proteins such as phytoglobin to regulate the homeostasis of NO by converting nitric oxide to nitrate. Then it can also bind to the cytochrome C oxidase to modulate the mitochondrial respiration under the stress condition. Beside this, it can indirectly NO signaling mechanism that interact with the reactive oxygen species, that is ROS, in which NO reacts with superoxide to form a peroxynitrite, which modifies and alters the signaling pathways. Beside this, NO that is binds with the cGMP and calcium dependent signaling, in which NO activates the guanylyl cyclase, that leading to the productive of cyclic GMP, which modulates the gene expression and enzyme activity, in which NO increases the intracellular calcium levels and triggering the calcium dependent protein kinases that regulate the various stress and development responses. So NO production occurs through the two enzymatic pathways as we have discussed earlier, and these pathways that lead to the S-nitrosylation, which is also act as a signaling agent in this particular figure. So as we have seen the biosynthesis pathways of NO, so it is very important to discuss the what are the sources of NO before discussing what are the roles of NO in growth and regulation, and how we use the NO in other agriculture practices. So if we see the sources of NO, nitric oxide is a crucial signaling molecule as we have discussed earlier. The sources of NO, which are donors which can be used for our research purposes. So NO donors are compound that release NO at are used to study in physiological effects, and in which the most significant and most used donor is the sodium nitroprusside, which is commonly called as SNP, which is one of the most widely used donor in plant studies and releases NO in aqueous solution. So this one is the most studied donor which is used in the physiological studies, that is the SNP. Beside this, that is another donors of nitric oxide like the S-nitroglutathione. This is a natural NO donor that participates in S-nitrosylation of proteins, and diethylene enonate, a rapid NO releasing compound that is used in controlled NO release studies. So these are the some chemical NO donors which we can use in our physiological studies or research purposes. So beside this, suppose you have used NO in your physiological studies, that NO you have to study a specific function, which are the function which is regulated by the NO. So you have to use also the inhibitors, because many functions are linked with other phytohormone signaling molecules. So to detect which function is regulated by the NO, we have to use the inhibitors, because if we use inhibitors, that function if that regulated by the NO, that will not going to happen. So inhibitors is also very important in research practices, because it inhibits the activity of that particular compound, and NO inhibitors which is commonly used as that suppresses the production of NO, like which include the NOS-like inhibitors, because we have seen in the pathways of nitric oxide synthesis, there are two main pathways of NO synthesis. That first one is the NR pathway and the second one is the NOS pathway. So the inhibitors like NOS-like inhibitors that inhibit the competitive inhibitors of NO synthetase like activity. So the enzyme which synthesizes the NO, that is inhibited. So the function which is related with the NO, that will be not appear in that particular test plant or your. So these are the some examples here listed here in this particular, which are showing that is the inhibitors. And beside this, there are several scavengers also used for the study in the research functions. That is, the scavengers are those compounds which remove the Na from the sources. So scavenger neutralizes the excess NO and helping in NO detoxification and regulating its signaling pathways. And the most important in which is used as a carboxyl PTIO, a widely used NO scavenger that react with NO and reducing its bioavailability. So I am not discussing all of them. You can use sources, and the most common source is sodium nitroprusside, that is commonly known as SNP, and there are inhibitors like NOS inhibitors and scavengers like PTIO. They scavenge the nitric oxide. So you would like if you would like to study on the NO in physiological function, you have to provide a donor that releases the NO. You can study it, and if you want to check which function is regulated by the NO, you have to use the inhibitors or scavengers to check that particular process. So that's why I have discussed this first. Then I am going to discuss about the what are the growth and regulation function which is regulated by the nitric oxide. A nitric oxide is a crucial signaling molecule in plants that plays significant role in various physiological and development processes. Its function ranges from seed germination to stress responses, like it promotes seed germination. Here the green one indicates the promoting factors and the red one that is inhibiting, and that both the arrows green and red shows that depending upon the concentration of the nitric oxide and the circumstance of environment. So it promotes the seed germination, regulates flower development, influences cellular production in roots, and prevents chlorophyll degradation. Additionally, NO delays fruit ripening and modulates the lateral and adventitious root formation, and also inhibit the senescence and suppresses programmed cell death. So all these functions, how NO regulates all these functions. The first starting from the seed germination, that we will also discuss in the next slide. NO promotes seed germination by breaking seed dormancy and enhancing reactive oxygen species signaling. So the signaling which is involved in the regulation of NO, it is also involved the crosstalk of AB and gibberellic acid, which we will discuss in the next slide. So it also promotes the seed germination, then it also promotes the root development. So NO influences the root architecture by modulating the lateral root formation and root elongation by the interacting the auxin and cytokinin. So you can see here NO is involving in crosstalk with many hormones as well as the signaling molecules. Like in the seed germination, it is crosstalk with the gibberellic acid and abscisic acid, and for the root development, it has a crosstalk with the auxin and cytokinin. Similarly, for the flowering and reproductive development, NO plays a role in flower transmission and pollen tube growth, and it regulates anther dehiscence, ovule fertilization, and seed sets. And beside this, in photosynthesis and chlorophyll synthesis, NO enhances photosynthetic efficiency by regulating chlorophyll biosynthesis and electron transport chain, and it also protects the photosynthetic apparatus under stress conditions. And lastly, the senescence and programmed cell death. NO delays leaf senescence by reducing the oxidative damage and modulating the gene expression, and it also participates in the regulation of programmed cell death under the stress condition. So these are the some growth and development of processes that is mediated by the nitric oxide. So these are the some processes, and here I am going to discuss only the one part, that is the seed germination. Here you can see in this figure, for the NO that breaks the seed dormancy, for this it has a crosstalk with the abscisic acid and gibberellic acid. What are the role of gibberellic acid? Anyone can tell? Seed germination through the enzyme alpha amylase. So, functional interaction of NO induces the ABA catabolism and promotes the GA biosynthesis. And this GA biosynthesis that leading to the dormancy, release and germination. And the here arrows and bar indicate the positive and inhibitory effect. You can see here this ABA catabolism is here. This NO is catabolism that regulate the catabolism and it promote the synthesis of gibberellic acid. So, the dormancy is breaking by the nitric oxide through this phenomena and they promote the seed germination. That is why it is very important in the stress condition also, because many of the stress that reduces the percentage of seed germination like salinity or drought. So, as it breaks the seed dormancy, so it also help in the elevating the stress responses like salinity or heavy metal, all these processes which decrease the rate of seed germination. So, I am not going to discuss all the processes in detail. So, now moving toward the next topic, that is the role of nitric oxide in abiotic stress tolerance. So, we have already seen NO is regulating many growth and development processes from seed germination to programmed cell death. And we have seen that seed germination is play a very important role. So, here in this figure, a generalized representation of versatile role played by the NO in plant during various abiotic stress conditions. Like the signaling of NO underpins a multitude of cellular processes that involve in the development and abiotic stress responses in plants. And under stress condition, oxidative stress is generated which is diminished by the NO by modifying the morphophysiological, biochemical and molecular processes such as functioning of stomata and transpiration under water stress condition, activation of defensive genes and antioxidant enzymes and accumulation of osmoprotectants and crosstalk with different phytohormones. These are the some modifications that is enhances by the NO which help the plant to suppress or elevate this abiotic stress condition. For example, the presence of salt has been observed to prevent the seed germination and resulting in the significant agricultural losses because it decreases the germination of the seeds. And we have already discussed this, as we know, germination of seed is first stage in the development of angiosperm, and wheat seeds exposed to salt stress can be effectively elevated by the administration of this NO donor like sodium nitroprusside. This is probably due to the enhancement of beta amylase activity which is directly linked with the seed germination, and during seed germination beta amylase is involved in the degradation of starch and facilitate the conversion of starch into the sugar which is essential for the proper seed germination. And the mechanism is we have seen earlier in this particular slide ki how NO crosstalk with the abscisic acid and gibberellic acid to promote the seed germination. So, in this way I am giving just one example how NO help in the elevating the salt stress by promoting the seed germination. Similarly, in this figure a mechanism of different stress and how NO can elevate different types of stress. So, NO plays a protective role in drought, temperature extremes and heavy metal stress also. So, it regulates stomatal closure, antioxidant enzyme activities and osmolyte accumulation, and all these things is done by modulating the signaling pathways. So, NO enhances plasma resilience and under the stress conditions, and all these role is done by, so NO interacts with the different target molecules such as mitogen protein kinases that activate the signaling molecules. It also interacts with the cyclic glucosylation monophosphate, cyclic adenosine diphosphorylase and calcium. So, these are the signaling molecules in which NO interacts, and as the protection against oxidative stress, NO can initiate the cascade of signaling event which is listed here and alter the expression of gene and also facilitate the re-establishment of cellular redox homeostasis. So, one example we have seen earlier, and here you can see it regulates number of signaling pathways that initiated by the NO, and in this way it generates a protective mechanism for the plant to withstand against different particular stress condition, either it is a heavy metal or drought. Plant can elevate these conditions during the due to the presence of NO. And here in this diagram, specifically heavy metal stress tolerance has been discussed. So, specifically interaction between heavy metal stress and NO can vary depending on the type of heavy metal, its concentration and duration of exposure and plant species. And as you can see in this particular figure, that NO at low concentration, it maintains the stability of electron transport chain and recovering the process of different PS2 activities, improving the MPK pathway, and also it increasing the different antioxidant defense genes like APX, SOD, GR and CAT, and that maintain in all total cellular homeostasis, as we have discussed earlier, which is very important for the signal transduction, because if the level exceeds, what will happen? It can cause cytotoxicity. So, at low concentration, it maintains this homeostasis that improves the photosynthesis and growth of the plant, that increases the yield. But at higher concentration, that disturbs the cellular homeostasis and ultimately inhibited the photosynthesis and decreased the yield of the plant. So, as we have discussed earlier, its concentration is very important and which is regulated by its homeostasis, which involve the biosynthesis and scavenging of nitric oxide. So, besides abiotic stress tolerance, NO is also involved in biotic stress tolerance, like NO also plays a crucial role in plant defense against pathogen and herbivores, and it induces systematic aqua resistance and enhances the production of antimicrobial compound. For instance, NO has been shown to protect potato leaves from Phytophthora infestans, a devastating pathogen which causes a much loss in the productivity of potato. Additionally, NO enhances herbivore resistance by promoting the synthesis of defense compound like antioxidants or secondary metabolites. So, it is also involved in the biotic stress tolerance. Then the next very important role of nitric oxide, that is in the nutrient use efficiency. So, nutrient use efficiency is essential for sustainable agriculture and it enhances the crop productivity while reducing the excess fertilizer usage and environmental pollution. So, how NO increases the nutrient use efficiency? First, NO use efficiency, like NO enhances the nitrogen uptake and assimilation by modulating the activity of nitrate reductase and glutamine synthetase, which are the two main enzymes of nitrogen metabolism pathway, and S-nitrosylation of NR increases the nitrate reduction to nitrite and promoting the nitrogen metabolism, which is one of the crucial steps for the nitrogen assimilation in the plants. Besides this, it also increases the phosphorus use efficiency, which enhances by it increases the phosphate uptake by inducing the expression of phosphate transporters genes, that is the PST, and NO mediated root system remodeling, like increase the lateral root formation and root hair growth that improve the phosphorus acquisition from the soil. So, as we have discussed earlier, that NO promotes the root and lateral root growth promotion by the crosstalk of oxygen. So, when the lateral root and root hairs growth are increases, so the nutrient use efficiency is already increases because all the nutrient is absorbed through the root hair with the water. So, when it increases the lateral root growth, it also increases the nutrient use efficiency. Similar way, it also increases the potassium use efficiency, like NO regulate the potassium uptake by modulating the activity of potassium transporters, that is HEC and CUF family, and NO protects plants from K plus deficiency, inducing oxidative stress by activating the antioxidant enzymes. And besides this, iron and micronutrient use efficiency is also increases by nitric oxide, like NO chelates with iron into making it more available for plant uptake and enhances the zinc and copper uptake by a particular metal transporter genes. And NO also play a role in elevating the iron deficiency chlorosis by increasing the iron mobilizing in roots. So all these nutrients, like we have discussed, nitrogen, phosphorus, potassium, iron, these are the very crucial for the plant growth of development. And NO increases all the efficiency of these nutrients, that is very important for the growth and development of the plants. So in the next, there are some study of case studies of NO, like several case studies have done on the nitric oxide. Here I have listed only few of them. Like in the drought stress, it is a major challenge in wheat which affecting the growth, yield and physiological processes, and nitric oxide play a crucial role in enhancing the drought tolerance by regulating the stomatal closure, antioxidant defense system and osmolyte accumulation and hormone signaling in wheat. So drought tolerance is involved in it regulate this particular process in several steps. Like first is role of NO in drought tolerance. It first regulated the regulation of stomal movements. So NO interact with abscisic acid to induce the stomatal closure and reducing the water loss. Then it activates the calcium dependent protein kinases which triggers the guard cell signaling and stomatal closure and enhances the expression of water channel proteins like aquaporins to improve the water retention. Then secondly, it enhances the antioxidant defense mechanism in the drought tolerant wheat plant, like it lead to the enhanced generation of antioxidant like superoxide dismutase, catalase, ascorbate peroxidase and glutathione reductase. And besides this, it is also play an important role in osmotic adjustment and metabolic regulation. Like NO promotes the accumulation of osmolytes like proline, glycine betaine, sugars which help in maintaining cell trigger pressure, and NO modulates this nitrogen metabolism by regulating the nitric reductase and glutamine synthetase that enhancing the protein synthesis and stress adaptation. So all these processes generally involve the crosstalk of hormones and gene regulation. And beside these wheat drought stress, NO is also enhances the nitrogen use efficiency in rice, like the donor of nitric oxide, sodium nitroprusside increases the nitrogen uptake and assimilation in rice and releasing the compound that enhance the photosynthetic nitrogen use efficiency, promoting the higher biomass production and improves the nitrogen retention and reduces fertilizer runoff, that contributing to the sustainable agriculture because it decreases the dependency on the chemical fertilizer because it directly involving the nitrogen use efficiency in the plants. So it lead to the sustainable agriculture. And in the third example, like we have discussed earlier, that application of nitric oxide as a donor has been shown to reduce the pathogen infection rates, like it enhances the PR gene expression and antioxidant activity and strengthen the cell wall structure that making plants more resistant. So these are some case studies, and if we see the interaction of nitric oxide with other phytohormones, so we have seen that most of the processes that NO regulates, that is by the doing the crosstalk with the other phytohormones or signaling molecules. Like example, it promotes the root elongation and lateral root initiation through the interaction with auxin. NO also regulates stomatal movement and drought condition by modulating the abscisic acid signaling, and all these interactions highlight the multifaceted role of NO in plant physiology. So in this figure, you can see that showing a different growth regulating processes and what are the crosstalk of NO doing with the different growths. So nitric oxide, as we have discussed, it involved in, like if you see NO and auxin crosstalk, NO promotes the root growth and lateral root formation by enhancing the auxin signaling, and this NO modulates the activity of auxin responsive genes such as pin form proteins which regulate the auxin transport that enhances the root hair development in response to auxin signaling. So with auxin, what it will do? It will enhance the pin protein that is involved in the auxin transport and that enhances the root development. If we see the NO and abscisic acid, NO acts as a mediator in ABA induced stomatal closure and preventing the water loss during the drought stress. And ABA triggers NO production, which activates the calcium dependent protein kinases that leading to the guard cell closures. Like example, NO required for the ABA induced systemal closure in drought conditions. Then if we see the crosstalk between the nitric oxide and cytokinin, NO modulates the cytokinin induced shoot development and leaf senescence, as cytokinin enhances the NO production, which regulate the cell division and differentiation, and NO interacts with CK signaling pathway to control delayed leaf senescence and chlorophyll retention. So, NO delays cytokinin induced leaf senescence and by influencing the antioxidant enzyme activity. With NO also interact with gibberellic acid, as we have discussed earlier in seed germination. NO enhances GA mediated seed germination and stem elongation by modulating the GA biosynthesis enzymes like GA3 oxidase or GA20, promoting the plant growth, and NO also reduces DELA protein instability, allowing GA to promote the cell elongation. Then NO is also have a crosstalk with salicylic acid, and NO and SA work together to activate plant defense responses against pathogen. Both induce systematic acquired resistance by upregulating the pathogenesis related genes, as we have seen in case of a biotic resistance. So, NO enhances SA mediated hypersensitive response and programs cell death in infected tissues. And if we see the crosstalk of NO and jasmanic acid, which is essential for biotic acid resistance like insect herbivory. In this case, NO enhances J-induced secondary metabolic production such as alkaloids and flavonoids. So, NO influences J-responsive gene involved in defense and wound healing. So, by this it increases the secondary metabolic production with the J crosstalk and increases the defense in case of a herbivory. And in the last, NO also have a crosstalk with ethylene and interact with the ethylene to regulate the fruit ripening, senescence and stress responses. In this case, NO inhibits ethylene biosynthesis by S-nitrosylating ACC synthase, a delayed ripening enzyme. So, NO with the crosstalk of ethylene, it delayed the fruit ripening. So, NO delayed the ethylene induced fruit ripening in tomatoes and other fruits. So, you can see it crosstalk with other phytohormone and regulate a number of growth and development processes in plants. Beside this, NO also play a very important role in agriculture. So, the accident application of NO donors, which we have seen earlier, the most common donor is sodium nitroprusside, has shown promise in improving crop resilience and yield. NO based fertilizer are being explored for their potential to enhance nutrient use efficiency and reduce environmental impact. Additionally, NO has applications in post harvest preservation, extending the shelf life of fruits and vegetables. So, as we have seen earlier discussed that how fruit life is increases because it delayed the fruit ripening by delaying the ethylene hormone. So, in the next one it also involved in some other activities like NO aids in the plant microbe interaction, promoting nitrogen fixation and microbial association. It also have a potential tool for pest management and fungal and bacterial disease control. Beside this, NO also enhances the crop adaptability to climate change and hydroponic systems. And NO also influences the production of phytochemicals which contribute to the plant defense and human health. As we have seen in the case of a herbivory, it interacts with the jasmanic acid and it increases the production of secondary metabolite that is crucial for the in case of a plant defense in case of a herbivory. Then it has an application in plant tissue culture that aiding the micropropagation and regeneration. Then it is also involved in the breeding strategies are exploring NO responsive traits for the crop improvement. So, these are some sections in agriculture where it is used for the production, uses in the agriculture for the better growth and productivity. And this figure showing all the details which I have discussed in this particular lecture from biosynthesis to the role of nitric oxide. As you can see here, this here, this showing the biosynthesis of NO, then this NO how it is scavenged, then its post translation modifications that is mentioned here. So if we see NO, as we have discussed earlier, synthesized by the enzyme that is nitrate reductase and NOS, and from the different polyamine pathways. Then by the signaling it increases the antioxidant defense system, and how when this antioxidant defense system is increases, the overall plant of the growth is increases under the stress as well as in the normal condition. So this diagram depicting the overall growth of the plant. So if we see the what are the challenges and future perspectives. So despite its potential, there are challenges in the precise regulation of NO levels and understanding its crosstalk with other signaling pathways. Although we have seen here many crosstalk, but still many pathways are also missing. So future research should be focused on developing NO-based agrochemicals and exploring its role in climate resilient crops. And these efforts could lead to the more sustainable agriculture practices and improved crop productivity. So in conclusion, NO is a versatile molecule with significant role in plants' growth, stress tolerance and nutrient use efficiency. Its potential application in agriculture are vast, ranging from improving crop resilience to enhancing nutrient uptake and reducing fertilizer dependency. So by harnessing the power of NO, we can move toward the more sustainable and productive agriculture system. So that is, thank you.
Thank you, Dr. Gauss-Chebasri, for your nice presentation in relation to one of the recent growth regulator, that is nitric oxide. So today's session is going to be end. So there is a feedback form which has been given to each and every student. So please submit it by tomorrow. All the students are requested to submit the feedback form by tomorrow. And once again I am telling that the attendance is the compulsory as per the guidelines. So please just sign on your in the name in front of your name, not to the other's name. It is not a good practice. If you are here, then you have to sign it. If you are not here, so don't do that, because it will be not a good practice and also it will be noted in the wrong way. So tomorrow we will meet again at 9:30 by one of the lecture of the Professor Andrzej Bajguz. Thank you so much. One more thing, that we have a validity session on the 14th, and and the certificate will be given in the validity section those who will be present to only.